I would like to welcome Dr. Yvette Taché again from UCLA to chair our first session this morning. Thank you, Dr. Taché.
So welcome everybody for this very interesting session on the mechanism of nerve-organ interaction. We have 4 distinguished speakers who have made really pioneer contributions how the vagus nerve, through its efferent and afferent pathway, regulate pancreatic endocrine secretion, immune function, gut luminal sensing, and the existence of neural crosstalk between the visceral organ. Each speaker has 20 minutes of presentation, followed by 5 minutes of discussions. So it's my pleasure to introduce the first speaker, Dr. Alexandro Sayer-Cedo, who is an associate professor at the University of Miami. And the title of his presentation is Autonomic Innervation of the Pancreatic Islet, a Cautionary Tale of Mouse and Human Studies.
Thank you very much. It's an honor and a pleasure to be here. I have to say that I learned a lot yesterday, and I learned that I know very little about these systems, and it's very, very important. So we are here to find out how peripheral nerve activity can be manipulated and simulated to improve organ function and also relieve medical conditions. And one of these medical conditions is diabetes. And central to diabetes is the organ I'm going to talk a lot, which is the pancreatic islet. It's central for the regulation of glucose metabolism, glucose homeostasis. And when this organ fails, we become diabetic. Now, I have to say that it's very, very difficult to study the pancreatic islet within the living organism, even in animals. And in human beings, probably it's almost impossible. So I won't be able, like you saw it yesterday, to show beautiful diagrams with arrows, and that were criticized, by the way, also. So our work is still in an initial phase. We're just trying to find out how the islets are innervated. We don't know how the islets connect to the brain, so don't expect that from us yet, but this is something that will come in the future. I'm going to show you some original data, so it's important for me to mention who performed most of the work that I'm going to show you. It's Rainer Rodriguez-Diaz, who is here in the audience, and you can ask him some questions afterwards. So what do the pancreatic islets do? They regulate one simple variable, which is plasma glucose. They have other functions, but the main function is to regulate plasma glucose, and in principle, it should be easy. It's not that easy, actually. And the real problem is that if this variable is not controlled, you get a condition, diabetes. If glucose levels are too high for a long term, you get diabetes. But what is also important is to avoid hypoglycemia, which can be fatal in the short term. So the pancreatic islet, by releasing insulin and glucagon, is able to maintain plasma glucose levels at around 5 millimolar, which is 90 milligrams per deciliter. So that's the major function of the pancreatic islet. Insulin is released to reduce blood sugar levels, and glucagon is released to increase blood sugar levels. So they are seen sort of like antagonistic hormones released from the islet. So what are the islets? As the name says it, they are islands of endocrine tissue that are dispersed in a sea of exocrine tissue in the pancreas. And that's one of the real problems with studying pancreatic islets, is that they are deeply, first of all, they're in the pancreas. It's not a very accessible organ. It's an organ that you cannot really touch. And second of all, they are only 5% or even less of the pancreas mass, and they are deeply embedded within the pancreas. So you can do some immunostaining on sections, for instance, here, and you can see the cytoarchitecture of the islet. You see a lot of beta cells that contain insulin here, alpha cells that contain glucagon, and there is a third important endocrine cell here, which is a delta cell that releases somatostatin. And that is probably important to regulate the other cells, the alpha cells and the beta cells within the islet. So despite their name, they are called islets of Langerhans. These are not islands, completely isolated islands. They are regulated by other functions in the body, and in particular also by the central nervous system. And this was recognized very early in the 19th century by Claude Bernard. He found out that by manipulating the brain, he was able to change glucose metabolism in animals. And a person that is very important for autonomic nerve studies is Pavlov. I think it's important to mention that he was probably one of the first ones who started manipulating sympathetic and parasympathetic nerves to see how gland function changed. So this idea has been around that the brain is able to change glucose metabolism, and the general notion is that if the sympathetic nervous system is activated, you get a reduction in insulin secretion and an increase in glucagon secretion. Now, when I say this here, it's important to keep in mind that the islets can work absolutely fine without the nerves. So after pancreas transplantation, for instance, where the pancreas is completely denervated, people receiving those transplants, they are able to have normal glucose metabolism. I think it's the fine regulation and the fine tuning where you have the contribution of the central nervous system and the parasympathetic and sympathetic system. And this type of regulation occurs in a physiological context that is called glucose counter-regulation. And this physiological mechanism is there to prevent hypoglycemia. So the general notion about the parasympathetic system is that if it's activated, you have an increase in insulin secretion, an increase in glucagon secretion. And you see this here mostly in the context of the cephalic phase, is when you're starting to eat. Before you're absorbing the food, somehow the brain is sending a signal to the islets to prepare them for an upcoming increase in nutrients, including glucose. So a lot of the information we know about how the autonomic nervous system could act on islets is derived from studies in which basically the neurotransmitters are applied on the isolated islets. This is just an example from our lab showing you that acetylcholine can stimulate insulin secretion from human beta cells, and it's dose-dependent here. So beta cells can be really stimulated by acetylcholine, which is, as you know, the major transmitter of the parasympathetic endings. So I think that what is missing here is information about the circuits. And what are the circuits? What are the nerves that connect to the islets? And I have to say that most of the work has been done with animals, and as you will see, the situation in human is very different. So I think I found this very interesting paper about a research roadmap related to what we want to do here in this workshop. And I think that one of the important steps, maybe the first step is to create a visceral nerve atlas, which includes two things: anatomical, finding out the anatomical circuitry, as well as the physiological, the functional circuits that manipulate the organs. And that includes probably also the pancreas. And this is something that we have been doing for the last 6 years, so maybe we can get some funding retroactively. I don't know. So if you now look at the pancreas section again, and you do again some immunostaining, you will see that you can see the islet just by its innervation. So we are using here 3 different labeling stainings. So one is for the whole nerves in green. Then you can see the parasympathetic endings in red. Here they contain the cholinergic marker VACT. And then you can see, it's more difficult to see around the islet, you see a lot of sympathetic boutons or varicosities. So they have a lot of nerve. Now, the problem is, okay, you can establish, you can quantify the contacts. What we have done also is to try to find out which cells are getting parasympathetic contacts. So we established some methods to do that, because the problem with a lot of the neuroanatomical studies is that they're very qualitative and they show just one beautiful image, but there are no numbers. But another issue here is, can we show that these connections are functional? You would say, oh, yeah, of course, there's a huge effect, but what type of effect and what context are these axons and nerves activated, and what do they achieve in the islet? And as I said in the very beginning, it's very, very hard to study the islets in the living organism. And what is even harder is to try to stimulate just the nerves that go to the islets. Yesterday, we saw a lot of studies where the vagus was stimulated. Probably when you do that, you stimulate everything down here, including the gut, which has a huge effect on islet function. So how do we do that? How can we study the effects of islet innervation? So what we do is something artificial, but we want to exclude all the confounding factors. And what we do is we place the islets in a region in the body where we can really manipulate the nervous input, and where the nervous input is really specific to the islets. And this is by putting the islets into the eye. And this is just an image illustrating the technique. We are able to isolate islets from mice, from rats, from humans, so we can put them back into the body, not back into the pancreas, but back into the anterior chamber of the eye. And this is now a rat islet that was transplanted into the rat eye. And what you see in green is the incipient revascularization of the islet 3 days after transplantation. Now, you can put enough islets into the eye to reverse diabetes so that these guys, you can see them, but in addition, you know that these guys are functional. They are responsible for glucose metabolism in the recipient mouse. What is very important here is that the beta cells, as shown here in the islet grafts, they are innervated by the parasympathetic axons that innervate the iris. And what is important, and this is the neat part of this experiment, is that you can manipulate the nervous input to the islet grafts by using the pupillary light reflex. So in the dark, you have mainly the sympathetic axons that go to the iris activated. In the light, you have the parasympathetic axons to the islet that are being activated. So just by using light, in principle, you should change the nervous input to the islets. So you are going to see now a recording from mice that have been transplanted with islets in the eye, and another group of mice that have the islets under the kidney capsule. And what we always notice is that the animals with islets in the eye, they had lower glucose levels, glycemic levels, than the animals with islets in the kidney. And in the beginning, we didn't know what until Rainer decided to switch off the light, which decreases the parasympathetic input to the islets. And at that moment, we found out that the glycemic levels went up. So that was already telling us that actually the nervous input is doing something. And when you switch on the light again, it goes down again. So that was telling us that maybe the nervous input is able to change insulin secretion. And actually, if we take these mice and you have them in light conditions and dark conditions, in light conditions where you have the parasympathetic input to the islet grafts, you see that the insulin secretion measured in the animal is also higher. There is another way to test islet function in animals and humans. It's by performing glucose tolerance tests. So what you do is you inject a glucose bolus in the beginning, and you challenge the animals with a lot of glucose, and then you see this glucose excursion over time, over 2 hours. And you measure the glycemia in the mice. So these are, again, mice that have islet grafts in the eye. And what we did is the test with and without light. So you can see that without light, the glucose excursion is much larger. With light, with parasympathetic input, the glucose excursion is much smaller. And that, again, is telling us that maybe you have increased insulin secretion here. Now, the question that we had is this mediated by a cholinergic input? And another neat feature of the eye is that you can just apply an eye drop. The eye drops that ophthalmologists use for patients. So we applied just a drop of atropine on the eye. And we did the same experiment now under light conditions, but in the presence of atropine. And what we saw is that it looked like the conditions in the dark, so without input. So this is sort of proof of principle that actually the nervous input to the islets is able to change hormone secretion. I think that had never been shown directly because you never were able to separate the effects of the direct innervation from the effects on other organs that the parasympathetic system has. So what I show here is that there is probably an effect of the nervous input here shown in red on the endocrine cell, and that tells the endocrine cell to release more hormone into the circulation. Looks beautiful. Let's find out where we can find the nerve to stimulate it. But the human islet is this one here, and this is the mouse islet, and there are huge differences. Not only in terms of cytoarchitecture, but also there are functional differences. You can already imagine that the paracrine interactions in the human islet are much, much more important than in the mouse islet, because here you only see beta cells, and beta cells only see other beta cells and not other endocrine cell types. But what is more important in the context of what we want to achieve here is this here. The mouse islet, as I showed you before, is highly innervated. You can recognize the islet because of its high density of terminal varicosities. The human islet is conspicuous because of the lack of innervation. So you can see in the immunostaining, you can see that the islet is this region here that is devoid of axons. Now there are some axons in the middle of the islet, and I can tell you right now that these axons are not cholinergic. most of the acetylcholine, surprisingly, in the islet comes from the alpha cell, not from the nerve fibers like in a mouse. And what we have as innervation within the islet is most likely sympathetic. And I'm showing you this graph here to show you that there are a lot of blood vessels within the islet that are that contain smooth muscle cells that we think, and I'm not going to show you the movie because of time, but we think that these sympathetic axons, they are mainly contacting the smooth muscle cells within the islet and not endocrine cells. So we have two different patterns of innervation. In the mouse, you have the sympathetic axons probably contacting the alpha cells, these parasympathetic axons contacting a lot of different endocrine cells. But in a human, it's mainly to the smooth muscle cells of the islet. So now how do you find out if this innervation is important for islet function? So again, we use the eye model where we can see the vasculature. In this case, we have a human eyelid transplanted into a mouse eye. You can see nicely the capillaries here. Now you can start looking at blood flow within the eyelid and try to find out if these sympathetic axons that go to the smooth muscle cells is able to change blood flow. So now we're going to go deep into the eyelid and look at blood flow. There's a movie here. Please focus on this region here and this region here. So the size of a beta cell would be this size here, and this is around the size of a capillary from around 8 microns. So you will see blood flow here, and the animal is breathing. This is a human eyelid again, and we are deep inside and opened here. So there is now blood flow in this region, and there will be also an opening here of blood flow in this region. So, is it coming? Oh, did you see? It opened there. So the possibility for local blood flow regulation is there in the eyelid, and we don't know yet if it's, we don't know yet if it's mediated by the nervous input. So this is a completely different way of how the nervous input could affect organ function. So now I will spend the last 3 minutes, trying to find out if, with the little information we have, if we can really manipulate autonomic nerves to improve glucose metabolism. So this is the big question. And I have to say that despite our data, I think there are some promising paths here. So one of the cells that I did not mention is the delta cell, and probably this cell is very important to regulate the other cells within the eyelid. And we have data showing that this cell is probably the one that is innervated by the sympathetic nervous system, in addition to the blood vessels. Now, of course, you could harness the parasympathetic system to act elsewhere, and that could improve eyelid function. That is a possibility. To increase incretin secretion from the gut, for instance, could be very beneficial for the eyelid. So instead of giving people incretins like Exendin or GLP-1 analogs, what you do is you stimulate the gut and you get your natural GLP analogs there. Now, it is clear that there's a tight relationship between the nerves and the blood vessels. So this is something that we can explore. And it has been done in part also in some studies in which the sympathetic nerves that go to the eyelids, they can be manipulated with tyramine, which is a substance that increases release of adrenaline into the bloodstream. So that has effect. It has been shown already in humans that it has an effect on insulin secretion. So this is another way of targeting the sympathetic axons. And it seems that maybe the sympathetic axons, in addition to having an effect on function, it could have a trophic effect. And last but not least, and this is sort of a transition to the next speaker, is there are sensory axons in the eyelid too, and maybe also the sympathetic axons that can interact with the immune cells within the eyelid. This is a whole new field that we want to explore that is also very promising, especially because there are several papers out there saying that if you get rid of these sensory axons, you eliminate immune cell infiltration and you improve or you delay diabetes in mice. And there's a new study also showing that if you get rid of all the sensory axons in the body, you can prolong life in mice again. And the way life is prolonged in the mice is by increasing insulin secretion, which we don't understand how, but that's what they find. So the issue here is that for all these studies, what we need, I mean, there are a lot of claims out there, but there is very little rigor. I mean, we need to do better studies, better anatomical studies, better functional studies. And what is really hard is to establish the functional effects of these nerves. So we'll have to be creative here. I mean, one thing is maybe doing slices of human pancreas and try to keep the nerves there and try to stimulate the nerves and maybe find out what the nerves respond to, especially the sensory axons. And of course, once we know more about those functions, another challenge will be to find adequate stimulation points. As we saw yesterday, I mean, maybe if we stimulate the vagus, we improve glucose metabolism. And maybe if we get rid of the sensory axons, we can prolong our lives. But the problem is that this is probably as unspecific as using pharmaceutical drugs. And the whole purpose here is to use the specificity of the innervation to have specific effects in the organs. And that's what we need to find out. Okay? Thank you very much.
Thank you very much for this elegant presentation. Any questions from the floor? We have a 5-minute discussion.
Yes.
Yeah, that was really very nice. Have you had the opportunity to evaluate blood flow regulation in the anterior chamber from normals and diabetics transplanted, and whether or not neural control of blood flow is different?
We will propose that in a grant, probably. I mean, it's something we want to do. It's something that is actually a major question. How does this change in diabetes, and how does the nervous input affect the islet, and can it be used to improve islet function? Yeah.
Dr. Bertu?
Very nice. So do you have some idea of where you would use stimulation here? So yesterday we talked all about these nerves. So where do you see a possibility to actually stimulate, let's say, specifically insulin secretion or inhibit insulin secretion?
I mean, we would have probably to stimulate the nerves that go to the pancreas, and that will stimulate also the exocrine portion. So I think it will be impossible to just stimulate.
What are these nerves that go to the pancreas?
Rainer, can you help me with that? I mean, and Mike, get the mic.
So we have the hypothesis that maybe stimulating the celiac ganglion have an important effect in both. That is the final effector for both systems. So, but we are working on that.
That's the final effector for what both systems?
For both, for parasympathetic and for sympathetic system. We believe that.
It's a hypothesis.
It's a hypothesis. We are working on that.
Because we are not seeing parasympathetic innervation. We're not seeing parasympathetic innervation of the eyelid. So we think that maybe the final part of the parasympathetic is mediated by the sympathicus. It's a crazy idea, but it's based on your findings.
Yeah. Well, it's... I will apply for the grant, too.
Interesting talk. I guess the question I have is, have you examined the glutamergic innervation of the islet in human?
With what type of innervation?
Glutamergic.
We have looked extensively at glutamate systems in the islet, and we don't find anything in nerves, but we find that the glutamatergic system, a lot of the components are present in alpha cells also. So a lot of the interesting thing is that a lot of the neurotransmitters are present in the endocrine cells themselves.
Have you mapped the sensory afferent innervation? You know that there is intestinal fugal neurons that come from the duodenum that innervate the pancreas, the function of which is not well understood. Dr. Patricia will talk about it, but how this input from the duodenum to regulate the pancreas, how do you envision this?
So I am aware of the papers by Kirchgessner that show that there is enteric innervation of the islets, and that is an additional confounding factor, and I think that it needs to be addressed too, but we are not doing it right now, but I think it's important.
Thank you very much. So it's my great pleasure to introduce the next speaker, who is Dr. Kevin Tracey, who is the president and CEO of the Feuchtgen Institute for Medical Research and director of the Laboratory of Biomedical Science. Dr. Tracey has made really a major breakthrough contribution in relationship with the regulation of inflammations, and he will present new information about the inflammatory reflex mechanistic approach to bioelectronic medicine.
Thank you. Good morning. We're a room full of professors here. What's everybody's worst nightmare? Following a brilliant lecture. So my nightmare is now being lived. I'm following a brilliant lecture, and So since that's the situation I'm in, I'd like to start by saying if there's a list for paying back earlier work, as Alejandro proposed, I want to be number 2 on the list after him for funding. So I am a neurosurgeon that is interested in inflammation for some 30 years now. When I was training to be a neurosurgeon at Cornell, an 11-month-old girl died in my arms of sepsis, literally died in my arms. And the tragedy is that we were getting ready to actually send her home. And I couldn't explain to the family what happened. The dogma then was that infection being present in her somewhere had killed her, but we couldn't find any infection. And we couldn't find any endotoxins or poisons. And so there was a real disconnect between what I could tell her parents about what had happened to her, that she died of some kind of poisons, and what had actually happened to her. And so I've been studying that question for the last 30 years, which is really what led me to the topic today of understanding how reflex control of these potentially lethal responses is a possibility for us to map, as Alejandro just said, to define new circuits that I think hold promise for therapeutic possibilities. So early on in the 1980s, we were very interested in understanding the concept that cytokines made by the immune system can be necessary and sufficient to cause disease. I call this the cytokine theory of disease. The cytokine theory of disease does not say that all cytokines are bad. The germ theory of disease does not say that all germs are bad. But in some situations, cytokines such as TNF, the one that I worked on the most in the 80s, IL-1 and IL-6 can become both necessary and sufficient to cause disease, whether it's acute shock like Janice had, or inflammatory bowel disease, or rheumatoid arthritis as we've come to know. The point is that cytokines didn't evolve to cause disease. They evolved to maintain health. Cytokines have a range of beneficial protective activities in host defense, in cell proliferation, in wound healing, and on down, and metabolism, and on down the list. So evolution has evolved critically important mechanisms that regulate the activity of cytokines to maintain this healthy balance. We now, in medicine, by identifying specific functions of these cytokines, and albeit some of these cytokines have functions that appear to be global. TNF has 1,000 activities that can be mapped and studied. But nonetheless, it's sufficient to develop antibodies against TNF to treat diseases such as rheumatoid arthritis, Crohn's disease, psoriatic arthritis, and the list is growing. In fact, it's growing so rapidly that this approach to medicine is now essentially a $50 billion market. $50 billion a year is being spent on targeting these cytokines with biologicals to treat the diseases I mentioned. This represents 5% of the world pharmaceutical industry. And it's a market that's 15, 20 years old. So it's a tremendous opportunity to understand further how to control this in a way that might be therapeutically beneficial. My entrance into this occurred in the early 90s when I wanted to develop a molecule to block TNF. And the molecule we chose to do is shown here. It's called, we called it CNI 1493. Somebody else named it cemapimod. I prefer our name. And this is a tetravalent guanyl hydrazone. It has 4 guanidinium groups separated in space. So to a macrophage or to a cell, this looks like an arginine, only it's a big bulky thing. And what we designed this to do was to block arginine uptake into macrophages. And it was pretty good at doing that, and this molecule ended up being a very good inhibitor of TNF released from macrophages. So we were very pleased with that. We wrote about 40 papers on this molecule and moved it into clinical development. One of the things we were interested in using this molecule for was for stroke. And so we were doing experiments injecting this molecule into the brain to block the TNF in the brain during a stroke. And it was during these experiments that a complete surprise occurred, a complete accident. As a control for these experiments, we began to measure TNF not only in the brain, but also in the spleen, which is a major source of TNF in the body. And what we observed completely unexpectedly was that the drug in the brain, picograms of drug in the brain, completely inhibited TNF in the spleen, and in fact throughout the whole body. And this made no sense whatsoever. We literally talked about this experiment in the late 1990s. We literally talked about this experiment for month after month after month in lab meetings and could not come up with anything. We did think that the only thing that made sense was that the molecule in the brain was doing something to turn off an off switch, and that the brain was transmitting this off switch to the innate immune system to suppress TNF, and it turns out IL-1, IL-6, and other cytokines too. So we began by saying, well, maybe it's the pituitary, but that wasn't the case, because when we removed the pituitary, there was still this inhibition of cytokines when we put the molecule in the brain. So then we thought, what could the nervous connection be? Might there be a circuit that would be targetable to control this? And we ruled out the spinal cord, and next we cut the vagus nerve. And when we cut the vagus nerve, what we found was that the molecule in the brain was no longer sufficient to inhibit TNF. We could repeat these experiments by cutting the vagus nerve at various points along its trajectory, and I'll show you more of that in a few minutes. But what we now had was the idea that the spleen, the liver, the downstream organs, they don't know if the origin of the action potentials was caused by a drug in the brain or an electrical stimulus applied directly to the nerve. So I went to the operating room and I got hold of a handheld nerve stimulator that I would frequently use to map the facial nerve when doing acoustic neuroma surgery. And we applied this nerve stimulator to the distal end of a cut vagus nerve and found that that was sufficient to block TNF in the spleen, in the liver, and in other organs. And so we proposed this idea as a cholinergic anti-inflammatory pathway and published this in Nature. Ludmila Borovikova was a postdoc at the time in the lab. And what the results show is that in the first bar, if you have a sham animal, the TNF levels are very low. In the second bar, if you stimulate the animal by exposing endotoxin, you get a significant increase in TNF. But if you cut the vagus nerve, there's a further increase in TNF. So the vagus nerve is exerting a brake-like function. And you cut the brake line and you make more TNF. And when you electrically stimulate the vagus nerve, you drive these signals into the spleen and other organs, primarily the spleen, and you suppress a significant amount of the TNF response. A suppression that's sufficient to prevent the animal from going into shock. So in this middle line is the animals that go into shock from endotoxin. They go into shock because the animal overproduces TNF. When you cut the vagus nerve, the animals go into shock faster, sooner. And when you electrically stimulate the vagus nerve, block the TNF response, the animals are highly protected. And so now at this point in the story, we were thinking, this is in 2000, we're thinking, why? There's no little man in the head that's controlling the brakes or the switches. So how does this work? So one of my heroes in science is shown here, Charles Sherrington, who won the Nobel Prize in 1932 for discoveries regarding the function of neurons. He discovered the reflex, and he named the synapse and about 100 other things. He was a genius. And his concept of understanding the organization of the nervous system is one of the most important books written in neuroscience. And what he proposes basically is that if you understand a simple reflex, you can understand how to assemble a nervous system. But that being said, there's no such thing as a simple reflex because they're all connected. So we heard a lot about complexity yesterday. There's a simple reason for that. So Sherrington defined a reflex as beginning with an initiating organ or receptor whence the reaction starts, a conducting nervous path or conductor leading to that organ, and an effector organ or gland cells or muscle cells. And so in thinking about what we were sitting on now, we were looking at a vagus nerve as a conducting path or a conductor leading to an organ, and an effector organ, in this case being the innate immune system where TNF is being produced, that's under the control. And the question then immediately loomed large when framed this way in this thought experiment. What is the initiating organ or receptor whence the reaction starts to turn off the TNF? And at that point, we became familiar with and began to collaborate extensively with Linda Watkins, who made a seminal discovery that the vagus nerve can sense inflammation in a very important way. So what you're looking at are animals that were injected with IL-1. In red is what happens if you put IL-1 in the abdomen of an animal, a mouse or rat. It gets a fever, or a human, by the way. Fever occurs, but in yellow, when Linda cut the vagus nerves, the animal didn't develop a fever. So this meant that the vagus nerve was providing the sensory input to activate the fever response, which was absolutely critical, of course, to homeostasis. Now, you could bypass this. If you put enough IL-1 in the animal, it'll bypass the nerve response, go straight into the brain, leak in through openings in the blood-brain barrier, and cause fever directly. But in low-level inflammation, the vagus nerve can sense this. And I will tell you that Professor Najima in Japan has published a series of papers showing the onset of specific neurograms after cytokine injection. And in fact, now in my lab and work I won't talk about today, we have a major program in mapping the effect of these sensory neurograms induced in response to specific cytokines. And that's being supported by NIH, DARPA, and GSK. So now we were able to actually formulate the idea that we may have stumbled into what we called an inflammatory reflex. An inflammatory reflex being activated by the presence of inflammation, and that what we had found by stimulating the vagus nerve was the motor arc, or the descending arc of this reflex. So we spent the last 15 years mapping this. And we've done that by multiple methods, neurophysiological, genetic, and biochemical, to define this pathway. And I'll just show you some of that data today. In these experiments, we were helped in a big way by the fact that we were tracking TNF. TNF is made very quickly in an immune response. It's an early product of activation of inflammation. And we were lucky because TNF, which we actually had to prove, 90% of the TNF that is made in the bloodstream of an endotoxemic animal is actually made in the spleen. The spleen has one artery in and one vein out, and so we could map the circuit. We did that by stimulating the vagus nerve proximally near the brainstem and making lesions, cutting the nerve at various points. And the pathway requires action potentials descending in the vagus nerve to the celiac ganglion and action potentials traveling in the splenic nerve into the spleen. And a lesion anywhere along that path, either surgical or pharmacological, impairs the reflex. And in all those cases, this leads to actually an overproduction of TNF. Now, inspired by Warren Grill's work in mapping such circuits using cervical vagus nerve stimulation, Warren identified myelinated A and B fibers that cross the diaphragm with a threshold of firing of about 500 microamps. And in looking at this more carefully, Yakov Levine has shown that these fibers, which are activated by the order of 400 to 500 microamps, mediate the signal to the spleen that inhibits TNF. If you now move into the spleen and look at them, work done by Mauricio Rosas, a graduate student at the time in the lab, you can look at the cells in real time making TNF. So what you see in the top two panels in red are cells that are stained red because they're actively producing TNF. You're looking at the cells that make 90% of the TNF that appears in the serum during endotoxemia. When you apply a vagus nerve stimulation either to an intact vagus nerve or to the distal end of a cut vagus nerve or to a splenic nerve, in all situations you block that production of TNF shown in panels C and D. So now the question became, these are, by the way, our marginal zone and red pulp macrophages. So now the question comes, well, what is the nature, the chemical nature of the signal that turns off, or the neurotransmitter that turns this off? Well, it's acetylcholine. It's acetylcholine because the receptor that's absolutely required on the macrophage to control TNF is the alpha-7 nicotinic acetylcholine receptor. And so work done here by Hong Wang, a postdoc in the lab, shows in the first panel that wild-type mice make TNF when you give them endotoxin. And wild-type mice in the second panel, given a vagus nerve stimulation or a splenic nerve stimulation, have inhibited TNF. But the alpha-7 knockout mice have two phenotypes. The first is they overexpress TNF because they've lost the braking effect of the inflammatory reflex. And second, they overexpress TNF even when you do vagus nerve stimulation. So this single gene product is absolutely required in the spleen to control the release of TNF. Now, many of you in this room know that we have a problem at this point in the story because the nerves in the spleen are not cholinergic. The nerves in the spleen are adrenergic. But I'll come back to that in a minute. So after the acetylcholine interacts with alpha-7, Ben Liu and a number of other colleagues in the lab, Peter Olafson, have significantly mapped the mechanisms of signal transduction, which I won't spend time on today. But suffice it to say that alpha-7 expression on mitochondria in macrophages, not the cell surface. Alpha-7 expression on mitochondria is critical by stabilizing the release of mitochondrial DNA and cytochrome C, which prevents the activation of the inflammasome as a mechanism for preventing the significant release of TNF, IL-1, IL-18, and HMGB1. So this is all worked out. We understand the molecular biology, inside of alpha 7. But the real question that I already framed to you is, where does the acetylcholine come from? Well, it turns out that many years ago, nerves to the spleen have been mapped to lymphocytes. And you're looking here at a green lymphocyte, and you can see a splenic nerve ending touching, an adrenergic nerve ending touching that lymphocyte. The lymphocyte is green because here it's a transgenic mouse in which GFP expression is linked to the transcriptional elements that control the expression of choline acetyltransferase. And what Mauricio and Peter showed in this paper, the discovery that we made here, is that these acetylcholine-producing T cells are required for the integrity of the nerve circuit I've been talking about. So if you delete those cells, and this has now been done genetically as well as simply in nude mice, you lose the signal. So this is an adrenergic nerve controlling the release of acetylcholine from these specific T cells, which is required to complete the circuit through alpha 7. So if you do this experiment in nude mice, for instance, on the right, vagus nerve stimulation fails to inhibit the release of TNF. On the other hand, if you take a nude mouse and you give it those green T cells and do vagus nerve stimulation again, you restore the pathway. So not only do those T cells complete the circuit in an intact animal, but they remember that they were stimulated, and you can transfer them to a recipient animal. They become vagus nerve transmitter mice that can actually mediate the signal in a naive receiver mouse and change the phenotype. So I don't know of any other examples in biology, maybe some of you know this, where you can actually start with an electrical signal in one mouse and transfer a cell carrying the information to a recipient mouse and recapitulate the effect of the electric signal in the first mouse. And so what we know at this point of the story is that the circuit is mappable to the level of molecular genetics and signal transduction, and potentially what we had hoped, of course, is that there's the possibility for therapeutic intervention. So my disclosure here is that I co-founded a company called SetPoint, which I consult for, but I'm not on the board of. And they did a clinical trial recently, looking at this question, can you block TNF in patients with rheumatoid arthritis to therapeutic advantage? And this was a trial done by Paul Peter Tak, Ralph Zitnik, and Frieda Koopman, and reported at the American College of Rheumatology in November. These are the first 8 patients that were reported. And you can see here, this is the starting baseline rheumatoid arthritis score. And after implanting the device, patients got significantly better. I will tell you that the parameters of stimulating the nerve in this device are on the order of what I told you before, 400 or 500 microamps, which we estimate stimulates 1% to 2% of the fibers of the 80,000 fibers we talked about yesterday that cross the diaphragm. So this has people very interested, especially the patients. The New York Times picked up on this story last May, and what you see here is a 38-year-old woman who was in Amsterdam and had failed steroids, failed methotrexate, and failed 5 biological treatments. She was unable to pick up a pencil, and she enrolled into the trial. This was in April of 2013. She had a significant clinical response to this low-level stimulation and is now riding her bicycle to the Dutch coast, a near 20-mile round trip from her home. After the implant, I started to do things I hadn't done in years, like taking long walks or just putting clothes on in the morning without help. I was ecstatic. When they told me about the surgery, I was a bit worried because what if something went wrong? I had to think about whether it was worth it, but it was worth it. I got my life back. This is an incredibly important story for us to think about in the Sparks Initiative because it raises a couple of important points. The first is, why did it work in her, and how can we harvest the specificity to do this again in larger trials? Her response, whether it's the clinical response on rheumatoid arthritis or the response to controlling her TNF levels, looks exactly like the mechanisms I've been talking to you about that we developed over 15 years. But how do you scale this up? We need to know more about the circuits, more about the specific innervation, more about the parameters to do this. And second, no matter how successful we are in what we know today, there's going to be adoption hurdles to what everyone in this room wants to accomplish, which is to use devices to replace drugs. So I will conclude with 2 final slides. One is that the sensory input of this, I think, is a tremendous opportunity. And those of you who haven't seen this paper, I urge you to read it. It's from Cliff Wolf's lab at Harvard. And what Cliff discovered, much to the chagrin of many people in the immunology field, not me, I think it's great, is that the immune system is not alone on the front line of host defense. When an infection occurs, there's two people on the front line of the host defense, the macrophage and the sensory neuron. And if you look at a sort of typical map, a high-density map of sensory neurons, every cell in the body is within shouting distance of a sensory neuron. And what Cliff shows in this brilliant paper, it's a full-length article in Nature, I urge you to read it, is that these sensory neurons are positioned to mediate axon-axon reflexes that actually regulate that macrophage sitting with the neuron on the front line of host defense. So you can't talk about an acute innate immune response. You can't understand it in vitro. We have to study it in line with understanding the role of the nervous system. I think we're at a critical point in understanding immunology in this light because, in fact, it's impossible to separate the response of these two major systems in the body that respond to threat and produce memory, the nervous system and the immune system. At every level, whether it's at the level of inducing the response, at sensing the response, at mediating the response, at the target of the response, or at the behavioral result of the physiological and animal behavioral result, at every step, these interact. And so the challenge I put to immunologists is to say, to continue to study macrophages and T cells in vitro without understanding these sensory things that are happening in parallel and in real time is something we have to think about. And finally, to quote from the Scientific American article that was shown yesterday, I think we are really at a unique juncture in the history of medicine, and I think that's what the Sparks Initiative speaks to. Simple reflexes are distributed across the whole nervous system, and there's trillions of synapses that mediate our health. And we have the research tools, although we arguably need more of them, to map specific circuits in a mechanistic and functional way in order to understand how best to harness these for therapy. Charles Sherrington ascribed the dominance of the human species on Earth because of the power of its reflexes and because of the ability for our consciousness to get out of the way when the reflexes were working. But I don't think he could have envisioned back then the advent of technologies that we have today to control reflexes, to keep inflammation in control. But I think that time has come. Thank you.
Thank you very much for this very brilliant lecture. Any feedback and question?
I was wondering if your vagal nerve stimulation, I didn't get the connection between the vagal preganglionic and the sympathetic postganglionic. Are the vagal preganglionics innervating sympathetic postganglionics that then release norepinephrine to activate the the nicotinic receptor?
So what we know is that there is sufficient activity in the pathway to turn off cytokines in the spleen if you drive action potentials down the vagus nerve, into the splenic nerve, into the T lymphocyte, which releases acetylcholine, which turns off the macrophage.
And the sympathetic innervation is required.
Well, I don't like to say sympathetic or parasympathetic. I know I probably shouldn't say it in this room. I have to get a food taster for lunch, but I actually don't talk about sympathetic and parasympathetic. I think that the vagus nerve has 80,000 neurons. I think you just heard from Alejandro some very interesting ideas about this. And I think that our job today with modern mapping techniques and modern recording and stimulating techniques is to map circuits. Mm-hmm. And I think we should be following specific neuronal subsets, whether they're adrenergic. Henry Dale, at the time he discovered acetylcholine, recommended that we not define nerves by their function. He recommended that we define them by their neurotransmitters and by their anatomy. And I think today, with our modern tools, we have a tremendous opportunity, particularly I think through the Sparks Initiative, to define neural circuits, to define reflexes that control specific functions that we're interested in, whether it's insulin release or TNF release. This inflammatory reflex is a prototype. There's going to be hundreds of specific neural circuits that interact probably in many, many different ways that may or may not fit into the textbook dogma of sympathetic and parasympathetic being in opposition. I don't buy that.
All right, forgive me. Adrenergic. Does a ganglionic blocking agent block your vagal activation, or do the ganglionic blocking agents also hit your nicotinic receptor?
You can block the inhibition of cytokine release by pharmacological targeting with either 6-hydroxydopamine or by ganglionic blockers, if you stimulate proximally.
Great. Thank you very much.
I'd point out that the terms sympathetic and parasympathetic don't define functions. These are anatomical terms. Parasympathetic are autonomic neurons that arise in the brainstem and sacral cord, and sympathetic arise in the thoracic. Then you go further to define the chemistry of those pathways and whether they're adrenergic or noradrenergic or cholinergic. So I think we can still use sympathetic and parasympathetic. They're anatomical. They don't define any functions.
I'm not going to lunch with you either.
All right, okay.
But I of course agree with you. But I do think the challenge and the opportunity today is to map specific circuits regardless of whether they conform to what we've learned anatomically. Because I think that the anatomic is a starting point, the neurophysiologic is the next point, and as we move down the substrate to identify the cellular response and the molecular genetic basis of these responses, this is a whole new world with new technology that we should be using.
Mm-hmm. So I was going to ask the same question as Carl. So it looks like the pathway, as you described it, are efferent neurons which come down the vagus nerve. They go to the celiac ganglion. They probably activate those ganglion cells through a cholinergic synapse. Then those ganglia release norepinephrine, which then activate the T cells. And then the T cells release acetylcholine again, and then those target the macrophages. So is there a parallel system for this neuroimmune interaction in the gut? Because we know the vagus innervates the gut has all of these immune cells that are involved in function. So do we have the same thing there as well?
Yes, and there's great work being done in Amsterdam by this, by Wouter. What's Wouter's last name, Roy? Wouter? Huh? Wouter de Jong in Amsterdam is doing great work on this. I'll tell you one that's more interesting than the gut, actually, and more surprising to me, is a cell paper from Professor Arima in Japan. And what he mapped is the fact that signals originating in skeletal muscle, traveling up the posterior column, through some black box in the brainstem, come down the sympathetic chain, exit the lumbar plexus, and mediate adrenergic signal transduction that terminates on endothelial cells that form the blood-brain barrier in the lumbar spinal cord, signaling actually through the chemokine receptors CXCL12. The net of this pathway is it gates the ability of pathogenic T cells to enter the spinal cord. So said simply, if you have a mouse with EAE and you hang it by its tail, it doesn't get EAE. Now that's what he did. Who the heck thought of that in his lab? I have no idea. But if you hang a mouse by its tail and give it pathogenic T cells, they will not enter the lumbar spinal cord through this. So that's another. Those are two more examples, I think, of circuits. And I would also advocate, those of you who are interested in mapping these reflexes, that you give them names. Because I think there's going to be hundreds, and the more specificity we can put on this, the more opportunity there will be for our device inventors and our device developers to make devices and parameters that will be specific to specific functional reflexes. I think that's where we have to go.
Yeah, just one other point. The epithelial cells in the urinary tract and probably at other sites also are detectors of bacterial invasion, maybe viral invasion. And those cells can release, among other things, acetylcholine. And acetylcholine then can trigger sensory nerves and evoke reflexes. And so the epithelial afferent interactions are probably involved in host defense mechanisms as well.
Absolutely. I think, as Sherrington said, we have to understand all these individual circuits, and they're all connected.
Hi, Kevin. Hi, Colin Reardon, UC Davis. We've been talking a lot about mapping. How does inflammation change these circuits? Do you actually see changes that start to develop? Is that going to be important for specificity coming later on? And what about variation between even inbred mice? I wonder if you could comment a little bit about that.
So the last one, I don't know. I don't know much about the neural differences in these immune controllers in the inbred mice, but all of the other areas are areas of active study, and they're incredibly important questions. So when one looks, for instance, at collagen-induced arthritis, there's overwhelming evidence that if you induce this is the standard model of autoimmune arthritis in mice, and when you induce the disease, the mice at first don't have any significant inflammation. They make antibodies against the collagen adjuvant, against the collagen antigen. In the period of time that precedes the onset of the arthritis, however, if you look in the spleen, the splenic neurons reorganize. They look like they die back. And so if this is a functional break in these mice, the brakes are failing in the lead up to the inflammation. Then the mice get rip-roaring inflammation in their joints, which resolves spontaneously over a period of days or weeks. And if you look at the spleen again, those neurons have come back. So I showed a picture of the green T cell with the red nerve, and I think that's a plastic synapse. So I think the opportunity for adaptability is tremendous. The lymphocytes are turning over, so you're going to have a pool of newly educated lymphocytes potentially repopulating these spaces, and the neurons are dying back and coming in again, and they could actually be now, quote-unquote, different neurons. So the opportunities for the kinds of questions you ask are incredibly important. I would go back even to a more simple question, which is, in none of these situations that I'm aware of do we even understand tachyphylaxis, sensitization, desensitization. We're talking about stimulating these circuits, and we need to actually map that functional component as well. I don't think we have a really good handle on that, and it's obviously important if you're going to treat people for a long time, whether it's arthritis, obesity, or whatever you're doing. We're going to have to figure out the responsiveness. What's interesting in that context about this circuit is that a very brief stimulation, 500 microamps in humans, 500 microamps, 1 to 2 minutes, shuts down TNF production in whole blood from the human donors for 24 to 48 hours. So once you turn these T cells on, they keep going. So we, I think, have had a major focus in the Sparks Initiative thinking about the refractory period, the responsiveness in the nerve itself. But to do this right, we're also going to have to deal with the biological half-life of the responding cells. If you need 1 or 2 minutes of stimulation a day at 500 microamps, it might be a bad thing to do it every 5 minutes at 6 milliamps, especially when the target's already doing its thing for 2 days. These are all important questions.
Thank you very much. It is now my great pleasure to introduce Dr. Ellen Raybould, a former colleague of UCLA, who is a professor at the Department of Anatomy, Physiology, and Cell Biology at the University of Davis and former chair of this department. Dr. Raybould pioneered the role of capsaicin-sensitive afferents in relationship with the effect of the vagal actions on gut function. And more recently, she shed a new light on the understanding of the mechanism through which sensory afferents pick up information from the bacteria and different components of the meal. And she will talk today about the plasticity of vagal afferent pathway, nutrients, and microbial drivers.
Well, thank you very much, Yvette, for that introduction. So I'm going to talk today about some work that's been going on in my lab, but also to try and introduce some other data that's coming out from other labs. I apologize in advance for this is not a comprehensive review, but really just to try and talk a little bit about talking about these visceral afferent neurons as if that they're always the same, from day to day or moment to moment, but how they actually have a lot of plasticity. So I was trained as a physiologist, and so that's always the way I look at things. And so I think about what is it the gut does. And so every day the gut, it takes in food, digests it, and absorbs it. And it is actually the largest immune and endocrine organ in the body, and has not only to regulate its own function, but also regulates feeding behavior and other behaviors, and also glucose homeostasis. But what the gut has to do to absorb these nutrients is in apparent conflict with its other function, which is basically to protect us from the outside world. So it's one of the largest barriers or interfaces between the outside of our body and the inside of the body. And so it has to balance these two seemingly opposing functions. And so I always think of the gut as largely a sensory organ, because in order to respond to what's going on in the lumen, it has to be endowed with numerous sensory mechanisms. And certainly, as has just been pointed out by Dr. Tracy, is that the epithelial cells are sensors, and probably all of these epithelial cells are able to sense some component of the luminal environment. And so this can be non-nutrient and nutrient, but there are, of course, specialized chemosensing cells, and these can be the endocrine cells, the enterochromaffin cells, and brush cells. And these have been known for a long time. They're in the mucosa alongside the absorptive enterocytes and the mucus-secreting cells. And of course, as Dr. Taché pointed out yesterday, we now know a lot more about these endocrine cells from these GFP-expressing mice, suggesting that they may have intimate contact with the other primary sensory cells in the gut, which are the primary sensory neurons. So we've been very interested in vagal afferent neurons in my lab for the last 20 or 30 years. And so I just put this up to remind myself, to remind you about how the vagus, this is work from Hans Bettu, how the vagus innervates not only the thorax, but also every organ within the subdiaphragmatic abdominal organs. And even though the vagus innervates predominantly the stomach and the esophagus and the first part of the small intestine, there is, via the celiac branch, an innervation to the small intestine and also to the large intestine. And so just in sort of to summarize some of what we've heard over the last few days, that the vagal afferent neurons innervates most of the gut. It terminates at all different levels, the mucosa and the muscle. And it's that these vagal afferents send information to the brain to regulate function via activation of reflexes and information to higher brain centers to regulate behavior. The adequate stimulus can be both mechanical and chemical. And most of this activity does not lead to conscious sensations such as satiation and nausea, but is not perceived and generates reflex function to control the gut. So now we come down to the sort of the more precise innervation of vagal afferents, and this is from a review by Simon Brooks. And what I'm going to talk about is that these mucosal afferents, as we saw from Terry Powley's very elegant work, that if we look at it from a morphological perspective, there are two types of mucosal afferents. Ones that are in the villi that branch and are located underneath the epithelial cell layer, but also some that really surround the crypts and don't penetrate the villus. And we don't really understand the difference. Even though we can recognize these morphologically, we don't really understand how these differ in terms of their function. If we look at the electrophysiological recordings, we know that these mucosal afferents are sensitive to light stroking of the mucosa. They tend not to be active at rest, but can be activated by inflammatory or other chemical stimuli, and they are insensitive to contraction and stretch. And of course, they're markedly chemosensitive, so they can be activated by gut hormones and other mediators. So there are, as we heard again from Terry Powley yesterday, many other different kinds of endings in the gut wall, both within the smooth muscle and within the myenteric plexus. So now I'm going to distill everything that I've just said about how there are different kinds of receptors or terminals in the vagal afferent neurons, both morphologically and electrophysiologically. I'm just a simple physiologist, and I now distill this to representing it as sort of one neuron, one pathway. Now, of course, we don't really know, are these the afferents that terminate in the mucosa? Are they terminating in the muscle? Electrophysiologically, how do we classify these? But we've done a lot of work functionally to look at these afferent neurons. So we do know that they innervate the gut epithelium and that they can respond to signals coming from the epithelium so to transduce changes in the environment in the gut lumen. So we get activation of vagal afferent neurons and changes in parasympathetic efferent outflow, which can change gut function, so change motility, change secretion in the gut. It also can lead to changes in the activation of the HPA axis and also changes in behavior, such as feeding behavior. Now, one of the things that we've been interested in is that these vagal afferent neurons express many different receptors for substances that are released in the gut. Many of these are true hormones like cholecystokinin. Some are more like neuromodulators like 5-hydroxytryptamine, cannabinoids, orexin, leptin, ghrelin, incretin hormones like GLP-1 and GLP-2. So a plethora of receptors are expressed by these vagal afferent neurons. And we've been really interested in trying to understand how these play a role in function. What is evident is that there is plasticity in the expression of these receptors and an expression of peptides by these vagal afferent neurons. So if we take the nodose ganglion out from an animal that's either fed or fasted and look at expression of various receptors, we can see that in the fasted state, the receptor for PYY, the Y2 receptor, is barely expressed. But if we take it from an animal where we have fed the animal, you can see a marked increase in expression of the Y2 receptor. In contrast, in fasted animals, there's high expression of the MCH receptor and the cannabinoid receptor CB1, and this is switched off when we take this from animals that are fed. So there's a plasticity. So these neurons will change their phenotype depending on nutrient availability, whether the animal is fed or fasted. Similarly, there are changes in peptide expression. So CART is not expressed. CART is a neurotransmitter that, when released into the NTS, is thought to inhibit food intake. It's not expressed in the fasted state, but is upregulated in the fed state. This is just a quantification here. And MCH is highly expressed in the fasted state and completely switched off in the fed state. So this is occurring over the course of sort of 30 to 120 minutes after feeding. And so we think about this as a plasticity in these vagal afferent neurons. Now, as I say, we don't know the targets that are being innervated by these cells. We're assuming that some of them are entering the gut. Whether they're mucosal, we don't really know. And I think that's one of the questions that needs to be addressed. Now, what's regulating this plasticity in vagal afferent neurons, one of the mediators is the gut hormone cholecystokinin. So if we look at the case where there's no food in the gut, there's no food in the lumen, and you give exogenous CCK, in fasted state, there's high expression of CB1, MCH1, and MCH, as I just showed you. Give exogenous CCK, and you switch the phenotype of these neurons to express Y2 receptor and CART. If there's food in the gut that will release CCK, you see the fed phenotype, expression of the Y2 receptor and CART. If you block the effects of CCK with the antagonist lorglumide, or you knock out the receptor, which we've also done in transgenic mice, then you see that it switches back to this fasted phenotype. This is primarily work out of Graham Dockeray's lab at the University of Liverpool. So if we look at it diagrammatically, you can see that these neurons sort of exist in what we think of as an orexigenic state during fasting, and then in response to intestinal nutrients and CCK, will switch to a fed state or an anorexigenic state, where you'll tend to decrease expression of things that will increase food intake and increase expression of receptors and mediators that will decrease food intake. Now, we have shown in our lab that this is dependent on the ability of these neurons to respond to leptin. They express the leptin receptor, and it's activated through canonical pathways for the leptin receptor, and this is required to potentiate the effects of cholecystokinin. We've also shown that these effects can be inhibited by the anorexigenic hormone ghrelin. And what we think is that the lack of these satiety signaling in the fed state will tend to will induce intestinal satiety and tend to decrease food intake. So what happens in obesity? We know that in obesity, that from both animal models, and there's also some work in human obesity, is that lack of these intestinal satiety signals can lead to an increase in meal size and hyperphagia. So we used a model of diet-induced obesity actually in Sprague-Dawley rats, which is a non-congenic strain. And the only reason that this is interesting is that we wanted to separate the effects of high-fat diet from the effects of obesity itself. So on a low-fat animals diet, these animals remain lean. On a high-fat diet, half of the population are resistant to the effects of a high-fat diet. So even in the face of ingesting a high-fat diet, they remain lean, or relatively lean, whereas others become obese. And these obese animals are typical of diet-induced obesity. They have an increase in body weight, an increase in adiposity, they're hyperphagic, and an increase in plasma leptin. But what is really interesting about these animals is that they no longer see CCK. So when we inject CCK into low-fat fed animals or animals that are resistant to the effects of a high-fat diet, we can have this acute inhibition of food intake. And this is completely absent in these diet-induced obese animals. Other groups have shown this. It's also been shown electrophysiologically that if you record from these vagal afferent neurons from diet-induced obese animals, that they no longer respond, or there's a diminution in the response to CCK. So here you can see this is work out of Mike Bayak's lab in Canada. You can see here that a recording from vagal afferent neurons innervating the gut. There's a decrease in baseline activity in these neurons. This is the response to the increase in activity of these neurons in response to CCK, which is attenuated in high-fat fed animals, and similarly the response to 5-hydroxydopamine. So there's a global a decrease in the excitability of vagal afferent neurons in diet-induced obesity. So what's leading to this change in the ability of these neurons to respond to intestinal signals? So if we look at our change in phenotype between the fasted and the fed, in this case, if we just look at expression of CART, which is down in fasted animals, up in fed animals. If we look at diet-induced obesity in the animals resistant to the effect of a high-fat diet, we still see this change in the phenotype of these neurons, which is completely absent in the diet-induced obese animals. So they can no longer respond to these satiety signals. Why is this? We've done a lot of work in our lab, and what happens is that these neurons become leptin resistant. Now, leptin resistance in hypothalamic neurons has been known for a long time. But what we have shown is that vagal afferent neurons become leptin resistant, and they become leptin resistant before you can detect leptin resistance, at least in our hands, in these animals, in arcuate neurons of the hypothalamus. So this is a fairly early event. Here you can see we've just measured leptin signaling by looking at phosphorylation of STAT3. Here you can see there's a robust phosphorylation of STAT3 in low-fat fed animals, somewhat diminished in the animals on a high-fat diet, but that remain resistant, relatively resistant to the effects of high-fat diet. And it's completely absent so leptin signaling is completely abolished in these animals that are hyperphagic and obese. And this is accompanied by an increase in SOX3, or suppressive cytokine signaling 3 in these animals, as we can see here. And at the same time as we saw this, we saw robust leptin signaling in the arcuate nucleus. So what's leading to this leptin resistant in vagal afferent neurons? So here you can just see that with the effect that we see in normal animals. And what we've shown is that you no longer see this switch. In high-fat fed animals, you get leptin resistance, and you no longer see this switch from the orexigenic to the anorexigenic phenotype in vagal afferent neurons. We've also shown that these neurons seem to increase their response to ghrelin. So what's initiating this leptin resistance? So we've done a lot of work on the microbiota, and that's distilled into this one box. I don't have time to go into that. But essentially, we see marked changes in the microflora. And we think that these changes in the microflora lead to changes in the epithelium, which then probably also lead to more changes in the microflora. But what happens is that we get an increase in circulating levels of LPS. This has also been seen in human obesity. These are very, very low levels of LPS, nothing like you would see in sepsis, and they're chronic. And what we think is that this is inducing leptin resistance in vagal afferent neurons to change feeding behavior so that there's no longer this intestinal satiety signaling, and you get larger meals, longer meals, and eventually hyperphagia and an increase in body weight. So vagal afferent neurons express the TLR4 receptor, like epithelial cells in the gut. We hypothesize that this increase in plasma levels of LPS would increase intestinal permeability, activate the TLR4 receptor on vagal afferent terminals, and induce leptin resistance. So what we did was we gave very, very low doses of LPS chronically to rats, and indeed we find that they gain body weight like high-fat fed animals. They're hyperphagic, so even in the face, these animals are ingesting low-fat food, so they become hyperphagic, and again, we see this lack of response to CCK in these animals treated for 6 weeks with very low dose of LPS. And similarly to high-fat diets, these neurons become leptin resistant. So I think what I've tried to show you is that these vagal afferent neurons will differ depending upon the physiological state of the animal, whether it's fed or fasted. And this presumably is associated with changes in nutrient signals coming from the gut, but also probably in the longer term in microbial signals that are coming from the gut microbiota. We have just looked at LPS, but I don't think that's the only signal coming from the microbiota. I'm sure there are many other signals, both beneficial and harmful, that are really changing the phenotype of these neurons, both acutely and chronically, and changing the effects on behavior. So one of the things, as I say, I sort of reduce the complexity of vagal afferent neurons to sort of like to represent it as a single pathway. But what we really don't understand is what are the associations or the relationships between what we know from the morphological studies, what we know from electrophysiological studies, and adding on what are the neurochemical phenotypes of these vagal afferent neurons that innervate the gut and that show this plasticity in their phenotype. And one thing I think, so that I think is one challenge we have, but I think it's tractable. I mean, I think it's something that we can address. One thing that we haven't talked about at all in this last couple of days is the fact that in these visceral neurons are lots and lots of glia, or they're microglia. And we know from studies that are going on in our lab and many other labs, including that of Keith Sharkey's and Michel Noinlis in France and Keith in Canada, is that these glia are very important in setting the sensitivity of these neurons. And in terms of what I've just told you about today in this change in phenotype, can this change in phenotype be reversed? Because if it can, then maybe this is some way of being able to change appetite and satiety regulation in humans. Because I think that, you know, if it was easy to lose weight, we would all be thin, right? But it isn't, because hunger and lack of satiety are very aversive. And I think that if we can increase satiety signaling and maybe decrease hunger or anorexigenic signaling from the gut, maybe we can help with the control of appetite. I think that there are some groups who are working on sensitivity of signaling from the gut to the brain in human obesity, people like Michael Camilleri, Christine Finely-Bisset in Australia. But I think that we need to do more in terms of imaging to understand if we, in humans, whether we see these same changes in the vagal afferent pathway in obesity. And finally, I'd just like to thank members of my lab who really do all of the work. I just get to come to nice places in the snow to talk about it. But I'd particularly like to thank Will de Latigue, who has been instrumental in a lot of this work, and the NIH for the funding for my lab. Thank you very much.
Thank you, Helen, for this very nice presentation.
Good morning. That was a very nice talk. I'd like to bring up something I asked yesterday when we talked about blocking the vagus and obesity. So this is focusing on the afferent signaling.
Right.
Satiety. Calories in, calories out is the issue. So from what I understand is when people reach a certain body weight, it's very difficult to lose it, even if you restrict calories in, which gets to metabolism,
right,
metabolic rate, those kinds of things. So I'm wondering about that part of the equation in terms of these afferents signaling to brown adipose tissue, those kinds of things.
I think that's a really good and important point. We don't really know enough about how these different signals might impact brown adipose tissue. But I think even, as I say, the ability to be able to maybe regulate sort of satiety signaling could be helpful. Now, of course, one of the challenges is that there are neurons in vagal afferent neurons that express leptin receptors. Not all of these express ghrelin receptors. And so one would want to be able to reset the sensitivity of those neurons to leptin, maybe by electrical stimulation in some way, but not reset the sensitivity to ghrelin. You actually want to damp down the sensitivity to ghrelin if you think that that's mediating anorexigenic signals. So I think that by looking at the phenotype of this very mixed bag of neurons, that maybe we can get a little bit more traction on what exactly it is we're trying to do.
Sure. So to follow up on that, are you looking in your studies with indoor calorimetry or things like
that?
Yeah.
Oh, absolutely.
Have you seen changes in metabolic rate, temperature, anything like that?
Small effects, but small effects may be significant.
Sure.
Yeah.
Great, thank you.
Yeah.
I'm sure everyone at this workshop is wondering, can you mimic the changes in phenotype in the vagal afferents with electrical stimulation or with blockade? Have you found any parameters that can induce that kind of change?
So we haven't done anything like that. There are probably people in the audience who know better than I do. I think, as I say, the issue is that we have such a mixed population of sensory neurons in terms of their morphology, their electrophysiology. I mean, if you look at just neurons that innervate the small intestine, seem to respond differently to leptin from the neurons that innervate the stomach, for example. So. Yeah, so personally, I don't know. I'm sure there are people who do.
I think it's just very important, to see whether, since we're talking about electrical stimulation, the vagus, does it work on that? So I think that would be a great place to look. If nobody has done it yet, it'd be great.
Yeah.
Very nice. Regarding your microbiota theory, have you actually looked at bacterial counts in the small bowel, and is it induced by, does the fatty meal actually change the ability of the bacteria to sort of sneak up into the small bowel? And I don't know about rats or mice, wherever you're working in, but do you think the diet comes first, or do you think the bacteria are already somehow present in these more obesity-sensitive mice or individuals?
So that's a good question, and we haven't really looked at that to sort of see, is there a difference in the animal's baseline microbiota? And I think that's an important question. We've done a lot of work on changes in the microbiota and epithelial barrier function in my lab, and we see very early changes with a change in diet, in the microbiota, in epithelial barrier function. And we're now doing some metabolomic screens to see what it is that's getting across from the bacterial metabolites that are getting across the epithelium that may be influencing not only these nerve terminals, but may be getting into the systemic circulation. So I think it's a combination of the microbiota and the diet. It's a difficult thing to tease out, because as soon as you change the microbiota, you change epithelial function, and as soon as you change epithelial function, you change the microbiota. So to try and determine sort of what's the real driver is a little bit more complicated than we originally thought. But yeah, we're doing, yeah.
Maybe a last question and a short answer.
Sorry.
From your immunohistochemistry, it looks like the plasticity is occurring in all the vagal afferent sensory cell bodies. Can you comment on
it? When we do the quantification, it isn't all of them, but it's around the same percentage. So we've done a lot of colocalization to show the neurons that express Y2 and COT also express the CCK1 receptor, also express leptin and ghrelin receptors, which is around 60 to 70% in rodents.
So that's clearly not just the sensory neurons innervating your tissue.
Probably not. Probably not. No. Yeah, I agree with that. Yeah.
Thanks.
Thank you, Hélène. So then, the next speaker is Dr. Jay Patricia, who is the director for the Center of Neurogastroenterology and professor of medicine and neuroscience at Johns Hopkins University. He contributes to the associate number of gastrointestinal disease to nerve. And today's the title of his presentation is Duodenal Pancreatic Innervation, a dichotomous neural system with implications from inflammation and metabolic disease.
Thank you, Yvette. If Kevin was worried about following one great talk, I have my task cut out after 3 such wonderful talks. I do disagree strongly with Kevin about one thing he said. Kevin, there's nothing more interesting than the gut, okay? So I have a very focused title, but it actually has broad implications. What I thought I'd do today was talk about dichotomous innervation of visceral organs in general, and then discuss the implications for pain and inflammation, metabolic syndrome, which may come as somewhat as a surprise to some of you in the audience, as well as how understanding this peculiarity of visceral innervation may inform us about the mechanisms of bariatric surgeries and other approaches for metabolic disorders. And then we'll talk about the needs. So I think today, and perhaps yesterday as well, when most people talk about visceral nerves, they talk about enteric nerves, those that are contained in the wall of the gut. But a lot of the emphasis, I think, has been on the sympathetic and the parasympathetic, which, as Dr. Teekroat said, are anatomical, not necessarily functional descriptions. There is yet another kind of nerve which has been mentioned, but somewhat more in passing, I think, and that is the spinal nociceptive neuron. And this is not, of course, confined just to the GI or visceral organs. It is a nervous system that innervates the entire body and is responsible, or is well known at least, for the sensation of pain. What is not as well understood is that this is also a neuroeffector organ, so it not only carries information centrally, but can mediate processes in the target organs locally, particularly inflammation in a process that we call neurogenic inflammation by the release of active neurotransmitters such as CGRP or substance P. Complicating the fact is that even though this is a separate nervous system, it is transmitted or carried in bundles that are shared with other nervous systems, particularly the sympathetic nervous system, and this, of course, has implications in terms of how we specifically modulate the activity in these nerves. Now, the viscera do engage in crosstalk. This is something that is intuitively perhaps known to us because there are several physiological events that we normally take for granted as inhibiting each other. Urination, defecation, coitus. It's very hard to imagine life in all 3 of these or even 2 of these were to take place simultaneously. So there is already some kind of crosstalk that's occurring. Now, how does this communication occur? Classically, we think about it as the central nervous system, but it also occurs, of course, at the spinal cord level, particularly crosstalk between viscera from different regions, but at the same spinal cord level. But what I'm going to talk about today is crosstalk that occurs within the same nerve. And this is really the concept of dichotomous innervation of organs. So according to this concept, there's a single nerve that splits up before it reaches its targets, one branch going to an organ, say the pancreas, and the other, as shown here, going to the stomach. Now, these are well known and actually have been described by others in this room, particularly Dr. de Groot and folks from his lab. But we know that pathological events in one organ can induce disease in another via neurogenic mechanisms. I've given some examples here. Acute cystitis can be observed in response to colitis, prostatitis, or endometriosis. It can lead to upregulation of sodium currents in primary sensory neurons emanating from the bladder. So if you have colitis, you can affect neurons in the bladder, etc. What I'm going to talk today, and this is purely for illustrative purposes, is dichotomous innervation between the duodenum and the pancreas. Again, just to recap, single nerve fiber emanating in the dorsal root ganglia, splitting up, one branch supplying the pancreas and another supplying the duodenum. How do we know this exists? We followed the same methods that a lot of pioneers in this field have done. We injected retrogradely transported labels into the respective organs. So here, for instance, we've used cholera toxin B labeled with two different kinds of fluorescent dyes. One injected into the duodenum and the other injected into the pancreas. And then you look at the dorsal root ganglia and you see where the overlap is. So if you look and quantify this, you can see that of all the neurons in the dorsal root ganglia that emanate from the duodenum, nearly 50% in some segments actually show overlap with the pancreas and vice versa. And this is not just anatomical overlap, there's also functional overlap, and you can assess that by measuring nerve activity, teasing out single nerves, and then stimulating the respective organs. So here, for instance, is a nerve that appears to respond to the pancreatic duct as well as the duodenal duct. As you can see, that this is a nerve that shares innervation with these organs, whereas some of them only respond to the pancreatic duct stimulation and not to the other. In fact, if you quantify this, about 50% of nerves respond to both the duodenum and the pancreas in terms of mechanical stimulation. So what are the implications of this? So one of the things that we were interested in is how stimulation of the duodenum can lead to pancreatitis. And this is a phenomenon that's commonly seen in a procedure we call ERCP. So we were interested in knowing whether manipulation of the papilla itself can lead to pancreatitis. And what we showed here in summary is that if you provide a noxious stimulus just to the duodenum, in this case, it required a combination of mustard oil, which is a TRPA1 agonist, and ethanol, you can actually induce pancreatitis in, which is the adjacent organ, not by stimulating the pancreas directly, but via the duodenum. So you can see that pancreatic edema increases, histology severity score increases, and myeloperoxidase activity increases simply by providing noxious stimulation to the duodenum. Now, how do we know this is neurogenic? We used a technique for ablation of sensory neurons in the duodenum using a chemical called RTX, or resiniferatoxin, which is its formal name. And this is a toxin that takes advantage of its receptor, which happens to be TRPV1. As you know, TRPV1 is a master transducer of noxious stimuli expressed in a subpopulation of C and alpha delta spinal nerves. And ablation of these fibers can be done either with capsaicin or RTX, which is about 200 times more potent. So this is a useful tool for us to use, particularly in focal application, because local application of this chemical will cause reliably denervation of TRPV1 expressing neurons in that particular organ. So when we apply RTX to the duodenum again, not to the pancreas, we can, in fact, in that previous model that I've shown you, significantly attenuate edema, histology severity score, and myeloperoxidase activity, suggesting that this, in fact, is some kind of a neurogenic reflex mechanism that we are interfering in. It's, of course, actually more complicated than that. I just talked about the duodenum and the pancreas. We've also done similar studies with the stomach. And so it's not just a dichotomous innovation, it actually could be a trichotomous innovation, if there is such a word. But the bottom line is that studies like this and studies like this have been done with other adjacent organs in the pelvic cavity as well as the abdomen, suggest that adjacent organs, such as the stomach, duodenum, and pancreatobiliary region, behave as a sensory neural unit, and it's almost impossible to try and dissect out their neural innervation on an individual organ basis. This has, of course, implications for neuromodulation, which is the topic of this workshop. Here, for instance, if you stimulate the stomach, you can actually you can actually prevent cerulean-induced pancreatitis. So you're not actually directly stimulating the pancreas, but simply the adjacent organ, and you can theoretically have, or at least in a preclinical model, have a beneficial effect. What are the other implications of this kind of innovation? So we've been very interested in neural regulation of insulin resistance and other metabolic issues. And of course, this field was revolutionized by the observations that patients after Roux-en-Y gastric bypass had a almost miraculous remission of their type 2 diabetes, sometimes within the first couple of days after surgery before they'd lost a single pound of weight. And this was further enhanced by a company that developed this bypass sleeve and showed that simply by excluding the duodenal lining from duodenal contents physically, you can mimic the effects of gastric bypass surgery. So there has been a huge amount of interest in trying to understand the mechanism of these effects, and I'm not going to go into details. But our hypothesis was that perhaps this effect of duodenal exclusion was being mediated by nerves. Some evidence for this had, in fact, come by others in this field who had used that same chemical that I talked about, RTX, systemically. And if you use RTX systemically, it causes a generalized degeneration of sensory nerves all over the body. But nevertheless, it does result in attenuation of hyperglycemia, here, for instance, in obese Zucker rats. So this is oral glucose tolerance test before RTX, and this is after RTX, comparing vehicle to RTX, and there's a significant decline in postprandial or post-glucose challenge hyperglycemia. In addition, this is accompanied by a decline in insulin. So we wanted to test whether duodenal nerves had any role in the glycemic status of the animal, and we carefully applied RTX just to the duodenum and then performed glucose tolerance tests after RTX application on the surface of the duodenum. And you can see that 1 week, 3 weeks, and 7 weeks, there is a sustained effect on post-glucose challenge hyperglycemia. And this is quite significant, about a 25% or more reduction, which is comparable to what you would see with an oral hypoglycemic agent. And this is not just a non-specific effect. If you apply this on the surface of the ileum and colon, you actually see no effect on hyperglycemia. To further understand whether this was a neural phenomenon or not, we did surgery on these rats, cutting the greatest splanchnic nerve at various points that are shown here. This is actually illustration made by one of my research associates. It's very hard to find these kind of pictures in textbooks of rat visceral innervation. But regardless of where you cut it in the greatest splanchnic nerve, you can have a robust decrease in hyperglycemia. So we cut it above the celiac ganglia, above the suprarenal ganglia, at the suprarenal ganglia, and just at the celiac ganglia, and you get the same effect. So if you recall, this is really where only the sympathetics and the spinal nerves are, suggesting that this is in fact a spinal sensory effect and not just a vagal effect. So there are many potential hypotheses to explain these mechanisms. This is just one illustration. We are pursuing many others. But if you go back to this dichotomous innervation that I showed you, then it is possible that there is a local axonal reflex that is originating in the duodenum in response to nutrients and then attenuating the pancreatic insulin response in some ways. And again, going back to what I'd shown earlier, we are able to identify nerves in the greater splanchnic nerve that are responsive to both pancreatic and duodenal stimuli. And we've shown that these nerves, in fact, also respond to glucose in the lumen. Much of this data and much more of the mechanistic part will be presented at DDW this year, so I'm not going to spend a lot of time doing this right now. I want to conclude by saying that spinal sensory nerves to the viscera play an important role in health and disease. I've talked to you about inflammation. I haven't talked to you about pain, but we have lots of data on this in pain as well. And then I put some intriguing data, which I can't fully disclose yet, about the metabolic state and its regulation by these nerves. So these then are therefore legitimate and potentially attractive targets for neuromodulation in addition to the vagus and the sympathetics. Multi-organ innervation is a characteristic feature of spinal sensory nerves, but this poses several challenges and opportunities for neuromodulation approaches. Where is the nodal point? Where are you going to put your electrodes if you want to achieve the effect? If you put it here, you're going to stimulate both sympathetics and spinals, but that's not the only problem. You're going to stimulate more than just one organ, probably two, if not three at least. So how to restrict the specificity of the effect will be a major challenge. And perhaps this challenge will not be overcome unless we do more research on the mechanism and the neurobiology underlying these effects. So again, to reiterate a common theme, I think, that many investigators have expressed at this workshop, we need better anatomical and functional mapping to figure out local CNS and somatic connections. I want to thank the people in my lab who helped with these very difficult experiments, the NIH for all the grant support they've given me, and also to GlaxoSmithKline in general for the bioelectronics initiative and also for specific grant support. Thank you very much.
Thank you for this very nice presentation, please.
It's very nice. Because these sensory afferents regulate global sympathetic outflow, one might expect them to modulate cardiovascular function when they're stimulated any place in the body, whether it's the GI tract or the heart or the bladder. So when you ablate these afferents with RTX, do you see changes in cardiovascular function, blood pressure, sympathetic nerve activity? And I think that's especially important in the obese model because many obese models have concomitant hypertension.
Yes, of course. Those are excellent questions, and they apply not just to this particular class of neurons, but almost anything that we do. So we have studied this in obese models, and we are able to reproduce this data. And we're also looking at the cardiovascular effects by a variety of means. I'm not in a position to disclose that just yet.
I thought this is very interesting, and you raised the big point of complexity here, Jay. I thought perhaps I'd add an extra level of complexity to this because that's what we need here. The intestinal fugal neurons also project into those celiac ganglia. You've talked about, you focus mainly on dichotomous, but I know that there's been some suggestion that after inflammation has resolved in the colon, for example, there's loss of those long-term loss of neurons that might project from the myenteric ganglia. So it raises the point of a possible role for those modulating that pathway by their effects in the prevertebral ganglia.
Yeah, thanks. Simon, I mean, of course, the Mayo Lab has done a lot of work on the intestinal fugal neurons. We think that by the surgical experiments where we actually cut very close to the spinal cord, by the fact that we used RTX that is relatively specific for TRPV1 expressing neurons, that this is a non-vagal, non-sympathetic effect, but does not exclude a role for the intestinofugal afferents as well as the local reflex circuitries that they participate in. So I think it illustrates, as you said, how much more complex this system is and how many layers we have to peel before we can be confident of reducing this to a physical device or embodiment.
It seems very valuable in the same animals in which you made injections in duodenum and the pancreas to compare how many dichotomized neurons, about 50% in the spinal ganglia, how many in the no-dose ganglia where the injection site's the same size and we can actually compare vagal afferent dichotomization, trichotomization, and in the spinal primary afferent neurons.
Yeah, I mean, I think that's part of the anatomical mapping that has to be done. I think others have talked about the vagus, and I think this is appropriate for the question that was asked to Helen as well. When she showed her the plasticity in the nodose ganglia, I think the question was not just how many, but whether this was coming from the stomach, from the duodenum, and perhaps the fact that there's so many of these means that they're really coming from all over the place, and it's really hard to say there's one particular organ that is where we need to direct our efforts in terms of stimulation.
This may be related more to what the idea of the SPARK program is about. So Irv talked yesterday about the same approach to target cardiac afferents, which worked very nicely. What you've done here with targeting afferents with TRPV1 works very nicely, and it seems maybe easier and more targeted than an electrode approach. So the question I have is SPARK based on looking at only at electrode approach to inhibit and stimulate nerves, or is this kind of approach something that is being considered as well? Painting risperotoxin on the duodenum seems pretty easy and works really well.
So I can't answer that question. I like everybody else this morning, I hope they support my research. And so I'm very much pro RTX. No, I'm just kidding. I think it actually, I'll leave it to the NIH folks to answer your question specifically about what SPARK is about. But I think it conceptually and philosophically raises the question about when we talk about neuromodulation, is this a end in itself, or is it a means to understand the mechanisms so that we can even further simplify the therapeutic approach? Perhaps small molecules or some other methods. I mean, this goes back to what I showed about gastric bypass surgery. It's certainly not an end to itself, right? But it started this chain of thinking that we can reduce this to a mechanism that is much simpler and easier for the patients to deliver. So I think that's discussion perhaps we can have in the next 15 minutes.
For example, you can use devices. The renal denervation story, there are companies developing catheter-based delivery of chemicals to target nerves. So that's a device-based approach, but it's an ablation approach.
Yes.
Permanent ablation approach.
But they're also, that's correct. And there's also inhibition and ablation that can be done with devices. So you're right.
Right, yeah.
I will now call all the speakers to come in front, and we could have a general discussion, and if maybe there is question or comments coming from the Twitter discussions. So Dr. De Groot.
A question for Jay Pasricha. If you apply RTX to one organ and destroy the afferent innervation of that organ, does it have an effect on the innervation to the other organ? For example, is there degeneration in the dichotomized pathway?
Yeah, so Chad, that's an excellent question. We're actually studying that right now with morphological studies in the pancreas after RTX application to the duodenum. So I don't have the answer for you yet, but I will in the next few weeks.
Because this is important, has functional implications. If you apply RTX and you think you're affecting just one organ, that could affect reflexes coming from the adjacent, dually innervated.
So we think that, at least our hypothesis is that that's the mechanism by which this is working, is either a short axonal reflex or a long spinal reflex. We don't know that yet, but we do think that application of RTX in the duodenum will affect innervation in the pancreas, not necessarily all innervation, but at least that is dichotomous in nature. And then the next question is, what does that dichotomous innervation do in the pancreas? Is it the islets that are being innervated, which would sort of make sense if you're seeing an effect on diabetes? But we know that it's probably more than that because we're also inhibiting neurogenic inflammation.
So you don't know what the chemical mechanism is for the afferent impact on the pancreas. Is this through activation of autonomic ganglion cells? Is it a direct action of peptides released from the afferents on the islet cells, something direct or indirect?
As far as the neurogenic inflammation is concerned, we've shown that this on the activation side, it's mediated by TRPA1. We haven't actually done antagonist studies to show whether the mediators are substance P or CGRP.
Do you think that the basis of dichotomizing synapses is also the base of what somebody showed yesterday about stimulation of this Chinese acupuncture regions? So, because if we are talking about that, it would be even simpler. So we are doing the research of the investigation that will validate thousands of years of Oriental medicine. So maybe you don't need to pain the duodenum, but maybe you stimulate something else, and then they go to the pancreas, what is known by experience. So Spark will become providing the scientific base of something that I haven't received any of this treatment of acupuncture in my life, but I know so many people that are successful with that. And with electrical stimulation of somewhere else, and then you have huge effect. But we can't be touching this in a real way, and that is my question. That could be
yes. So I think the purpose of this talk was to show that there are many levels at which the nerves can talk to each other. It's not just through long reflexes, which are very important, as Kevin has shown, but there's all these short circuits that are occurring. And we need to figure out how to understand them so that we can exploit them better. But specifically, it is not just also visceral communication. It has been shown a long time ago that a single afferent neuron from the upper limb can branch off and innervate the heart. So that may provide another explanation for why heart pain is referred to the arm or is felt in the arm. Traditionally, it was thought it's only at the spinal cord level, at the level of the second order neuron, but it may actually be occurring at the periphery as well. So all of these have to be factored in when we start peeling the layers off, as I said.
Any other question? Maybe I could ask Ellen, in relationship with Dr. Jeff Gordon, work on the obesity and use in the modulation of the bacterium. What is your overall thinking based on your own experience and how you could relate to some nerve mechanism? It's just like a very broad question.
So I think that it's turning out to be a lot more complicated, as these things are, because the more the sort of new high-throughput sequencing like MySeq are being used to study the gut microbiota, then we realize that we have to get down, we can't just look at phyla level changes, but we have to get sort of down to family and genus level changes. What we've tried to do is because we're sort of interested in what the onset of the leptin resistance is to sort of say, well, what's happening before the animals become obese? What are the initial changes in the microbiota and the gut? And then what happens when the animal is obese? And so those are also very different. So it's very time dependent as well. So that's sort of, I think, where the field is going in that respect. And then I guess the other question that people are looking at is, can you do something to actually to sort of get the microbiota back to where it should be? So if you have a dysbiosis, if you reverse that dysbiosis, can you and send it back to a more normal level? Can you change some of the changes in function that you see? So I think that's one thing that I think is important. And then to try and understand, how the microbiota, all those commensal microbiota are really, producing both beneficial and harmful effects outside of even being pathogenic, obviously.
The fact that the vagal innervation, where leptin is acting, is very small within the distal part of the colon. How you envision the nerve interactions?
So we've been looking at the changes in the gut microbiota along the length of the gut. And sure, in terms of the numbers of bacteria, there are far more in the cecum and the colon. But there is a microbiome in the small intestine, and it does change, and it changes very rapidly with changes in the diet. And so I don't think that I think there is a basis for thinking that there may be direct interaction of some of these vagal afferent neurons that are innervating the more proximal gut with the microbial compounds. But we're also very interested in what's happening systemically too. So what's getting out of the lumen from these bacteria that may be acting systemically.
Thank you.
I have a question for Kevin. Kevin, your work has obviously been pioneering in this respect, and I don't know whether you started off by trying to come up with a device or trying to just understand whether there was a mechanism that you could exploit pharmacologically. But I had a question about the pathway that you showed regarding the activation of T lymphocytes by adrenergic input and then subsequently acetylcholine. Now, that can all be mimicked, of course, pharmacologically. And the question is there something about the splenic microenvironment that makes that special as opposed to manipulating it pharmacologically? And you said 5-minute activation can result in long-lasting 24 to 48-hour changes. Is 5 minutes enough for the lymphocytes to go through the spleen and show that effect? I'm wondering if there's a much more generalized effect that you're talking about.
So the simple answer to your first part of your question is I wasn't interested in drugs or devices. I was interested in identifying therapeutic targets. So I'm agnostic to drug versus device. I think in the context of advantages or disadvantages, the more I've learned about it, if we have devices that can target very small, discrete components of a neural circuit to produce the therapeutic effect, I think there's tremendous advantages of devices over drugs. I think that you can think of a nerve as a drug deliverer or a drug delivery system, and there's one that has no side effects, potentially. If you could, there's no evolutionary pressure on these circuits to retain any toxicity. So one can imagine using a device to deliver the drug to a tissue where it's needed, nowhere else, for a short period of time as needed. And so there are, I think, theoretical advantages. And time will tell whether we can develop them in a way that devices will replace drugs. I think devices will replace some drugs, not all. And finally, I think in terms of the half-life question you alluded to, yes, very, very short stimulation. These T cells that I showed you we're in the process now of publishing a much more exhaustive description of the ontogeny and of the ontogeny and specific ontogeny of these cells. And suffice it to say that the expression of chat by these T cells is a specific subclass of all T cells. And these cells represent about 2 to 3% of spleen T cells and 2 to 4% of the T cells found in the circulating blood. So yes, activating this for a brief pulse leads to persistent release of acetylcholine from the spleen for 24 hours or more. And it's possible that, that this will have important implications on how to design a device with the right half-life.
So I think it's time to close this very interesting session, to thank the speaker for their outstanding talk, and we could have now a 30-minute break, and we could reconvene at 10:30. Thank you very much.
I think we'll get started and try and stay on time. You may have noticed there's, I think, a typo in the program which suggests that we would have two 20-minute discussion sessions. Actually, we only have time for a total of 20 minutes discussion. And we have 4 speakers, 10 minutes each. 20 minutes discussion. We'll take some questions after the first 2 and then some further after the total of 4. As you see, it's a session entitled Cross-Cutting Technologies. And I'm reminded of something I heard from Susan Hockfield, whom some of you may know, who did her PhD in neuroscience, I believe, at NIH, and went on to become the first life scientist who was the president of the Massachusetts Institute of Technology. And she talked about the way in which major advances have sometimes happened in science as a result of technology, particularly when people define new toolkits and parts lists. And she pointed that out, for example, in atomic physics a century ago, how it's happened in the chemical, in chemistry as well as physics, leading to the pharmaceutical industry, how it's happened in molecular biology, how it's happened in microelectronic industry. As we get new parts lists and new toolkits, we see substantial advances. So as you listen to this morning's speakers, think about the tools and the parts that they're describing and how you might be able to borrow these or how you might contribute tools to the toolkit. Because I think this has to be a collaborative effort with both technical tools, physiological, chemical, and conceptual tools to really make a leap forward in this area. First speaker is Viviana Gradinaru, who is now at Caltech, but previously did her PhD at Stanford with Karl Deisseroth on optogenetics and clarity. Viviana?
Thank you, Dr. Percy. Hi, everybody. Can you hear me all right? I'm doubted and also humbled by what we're trying to achieve here, just by looking at these simplistic diagrams. And I come from a neuroscience background, so forgive me if I get the terms and the details somewhat inaccurate, but I'll try for simplicity's sake to just frame our technologies without regard to a specific nerve or a specific organ, because I think that a useful tool is a tool that can impact many different areas and many of the pathways that we talked about yesterday and today as well. This list here of desired parts is something that was said again and again by the previous speakers. So there's nothing new here. On the picture, though, I want to highlight what Dr. Creasy said, that tools, part lists, and technologies do facilitate advancing knowledge. And what you see here is an overlay of action potentials. Let me see if I get the pointer. Right here. So electrophysiology as a basic method to record neuronal activity, and it's overlaid on Golgi staining, a way to do detailed anatomical mapping of nerves. Together with other technologies, so having these modalities in hand really facilitated understanding the circuits at hand and also informed ways to interface with the system to achieve both anatomical and functional knowledge. And the hope is that we can use some of these tools and also some new ones for the peripheral system as well. And I've heard the need for better wiring diagrams at the different target organs. We also need a good genetic makeup of the innervated cell types. So in terms not only their shape and location, but their subtle genetic identity and composition. And we need the functional maps as well. Anatomy is just a diagram, and without functional, we will fall short in our knowledge. And hopefully, 1 to 3 can inform interventional strategies. So to sum it up, neural circuit correlates of function and dysfunction will generally help the task at hand, which is to intended, as I understand it, to perturb peripheral nerves to better their target organs and the organism health. Today, I will focus on 3 technologies that hopefully can be enabling, and these are, the first one will be tissue clearing for anatomical and phenotypic mapping. The second one would be optogenetics, but broadly defined. This also includes chemogenetics and modalities to achieve control of specific cell types with high precision. And this can be used for functional mapping. And also to facilitate such mapping, both anatomical and functional, at least for animal models, we do fall short in our ability to target sensors and actuators to peripheral nerves. So further work on viral vectors to deliver such probes would be needed. Because of the short duration of the talk, I'll go straight into some case studies of what we can achieve with tissue clearing or optogenetics or viral vectors. And what you see here is an example of how tissue clearing can facilitate mapping through very large volumes. Here is a rodent just perfused and not cleared, and then through a series of steps, you can achieve a well-transparent organism. And the way you can do that is first to diffuse in monomers for a hydrogel to provide structural support. You can cross-link and form a mesh, a tissue hydrogel mesh, that then allows sufficient support for detergents to come in and remove the lipids while still preserving the shape intact. This detergent can be washed out with buffers, and now by using the same vascular routes, you can diffuse in probes. You can diffuse in small molecules. You can diffuse in antibodies to achieve not only rough anatomical mapping, but also understand the identity of cells and map neurotransmitters and where they are located. This method does preserve endogenous fluorescence. So what you can see here is that this can achieve clearing across a variety of organs of interest to this meeting, and we've heard beautiful talks about most of these organs. And embedded within these transparent maps are highly interconnected and complex nerves. So just to show an example how one could visualize such nerves. For example, by delivering systemically, you can use AAV9. It's a serotype that when delivered systemically can achieve broad transduction of cell types, including nerves. Here is shown at a low dose. And what you can observe is, for example, a nerve here for the muscle and also for the pancreas. You can achieve cell type specificity by either promoters or Cre lines to further narrow down your nerve of interest, and then you can follow it through. This is applicable to many organs, and you can see here examples from the adrenal gland. And I will highlight here what it looks like a cell body with processes. Of course, because the method allows antibody staining, one could then go and reveal the neuronal nature of such processes and also try to identify what kind of neuron this is. And same goes for the other organs as well. The method is not limited to rodent tissue, though. You can apply it on biopsies or on fresh tissue from clinics. And here is an example, I think following on the first talk in the morning, of cleared pancreas. This is you can see the scale bar here, 5 millimeters of volume of human pancreas cleared. And you can see here the extent of the problem. So at least for human tissue mapping, we confront ourselves with a needle in a haystack problem. They're very large volumes, and sometimes we need to point to a single islet or to a nerve going to that islet, and conventional histology is difficult to achieve this kind of data with conventional histology. So you can see here the distribution of the islets in the human pancreas. Now, this points to needs. So this talk is focused on where we fall short in terms of technologies. These technologies are new, could be promising, but if we don't amplify them, we're not going to be able to make full usage of them. One big problem with this technique is the availability and the cost of antibodies. Antibodies are large probes. Their diffusion through large volumes is still a big impediment, and the cost as well. So we've been looking at ways to bypass such problems, and one solution might come from camelid, from nanobodies that are much smaller. They have a smaller form, they diffuse better. And we've tried them in rodent tissue, so we infuse nanobodies to the vasculature, and you can see here this is against GFAP, and you can see the highlight of a blood vessel here. So better development of the nanobodies would really facilitate our understanding. A big problem, though, even with mapping and good nanobodies, remains tracing. We are dealing with nerves that span entire organisms, and this is a very difficult problem to have the start point and the end point and the route as well with all the collaterals. So we need to think hard about what we're going to do about data analysis and track tracing. And there's some work and some software available, but more is needed in this respect. And I just got the 2-minute warning sign, so I'm going to give a crash course in optogenetics. We've heard about electrical stimulation and the wonderful things it can do for patients. It's difficult to understand how it works because of its non-specificity. So methods that try to achieve the speed of electrical stimulation and the specificity of chemical modulation were developed, such as optogenetics that uses light and light-sensitive molecules, or chemogenetics, where actuators can be modulated by drug application. And the specificity is obtained by a few means. One is by having a promoter or intersectional strategies, but you can also achieve specificity, more important, by your site of stimulation. So in terms of peripheral nerves, you can illuminate the cell body or you can illuminate the axon terminus. And very importantly, you can achieve inhibition. So optogenetics is rather mature in the CNS field. However, very little work was done in the PNS. And I want to highlight, this is published work, so you can look in depth afterwards, but I want to highlight work from the Delft lab at Stanford, where transgenic chanorodopsin mice were used to control the sciatic nerve and muscle activity. And this highlights a need for optical cuffs if one is to have efficient light delivery to the peripheral nerves. Electrical stimulation does achieve modulation of the nerves. What's very difficult, though, is to achieve ablation or inhibition of the nerves. So this is where I think this figure, also from the Scott lab, is very important because it shows true inhibition of nerve activity of the sciatic nerve. However, there are the challenges of light delivery and also of the opsin delivery. This is work done in transgenics. And this takes me to, in a way, the last part of the talk, which is how do we have delivery vectors for specific nerves of interest. Jay mentioned earlier retrograde tracers. We would like something similar that has an ability to deliver either sensors or actuators. And here's an example that AAV6, AAV serotype, can be injected into the target organ and then achieve expression in the nerve for either imaging purposes, for pathway mapping, or for control purposes. And this is just a colocalization of the retrograde dye and the retrograde virus. To sum up some of the features of optogenetic neuromodulation versus electrical, there's specificity. You can activate or inhibit. The possibility for multiplexing could be there if the appropriate opsins are used. However, I should highlight the many difficulties as well, which are in terms of in terms of achieving light delivery to such nerves. So this is not trivial. So that's where methods such as chemogenetics might offer more tractable options for translations. For basic research, though, these are some points that need thinking and improving. Retrograde trafficking by viruses or other means. The opsin does take a very long time to traffic down the nerve, so the timeline of such experiments is rather slow. That's why it takes a lot of effort to get such experiments off the ground. Redshifted opsins, unfortunately, we have a few very good opsins now from the Boyden lab that achieve better penetration in the tissue, and also light positioning. So a lot of effort goes into miniaturizing such devices for mice, but we might not need them to that extent of miniaturization for actual human applications. So I'll close with my thinking and hope that the tools for discovery need not be the same as the tools for treatment. And we've heard beautiful case studies of electrical and chemical modulation working very well for treatment. So I'll close here.
I think, unless it's just a point of information, we'll move along. Is it just a point of information? But I think discussion will perhaps hold in the interest of time. Kim Butts-Pauley, also at Stanford, a professor of radiology, and will be talking about some tools in MRI and ultrasound. While the connections going, I think that's an interesting point that Vivian just made about the tools for discovery and the tools for treatment. I think we should bear in mind that the different toolkits.
Great, thank you. I'd like to talk about focused ultrasound today. I'm going to start with the brain. I think it has a couple of useful points I'd like to make, and travel to the peripheral nerves and then to systems, which is purposely left a little vague. Okay, so what is focused ultrasound? The idea is use a large area transducer outside the body. We can focus geometrically, electronically, achieve very high amplification, and achieve significant intensities deep within the body without any effect in the intervening tissue. As you saw here, this whitening within the gel and nothing in the intervening gel. So what I'd like to go over today is the use of focused ultrasound for blocks in the brain and peripheral nerve, for stimulation in the brain and peripheral nerves, and then stimulating organs or systems. In terms of blocks, I've organized this into permanent blocks with high heat and then reversible blocks with a low heat. And just to give a little bit of background, so focused ultrasound has been used or been looked at for many years. So this is a picture of one of the Fry brothers in 1960. So he was looking at treating Parkinson's patients by creating a permanent block in the brain. This is what a modern system looks like, uses 1,000 elements, achieves very high amplification. And the key here is that we have image guidance so that we can focus the ultrasound into the brain and get a picture of our focal spot. And this is being used right now for treatment of essential tremor. So many people have tremor. I'm tremoring right now, but it's not because I have essential tremor. I'm just a little nervous talking to you. Okay, but this is what happens. And then we can ablate the VIM nucleus in the thalamus. And then immediately, even while they're still in the magnet, we can get immediate symptom relief. And so this is what it looks like with a patient who's having a difficult time with a simulated eating task. And then after the treatment, then he's doing much better. Now, again, there's a reason why I wanted to show this to you. I'll come back to that in just a moment. Interestingly, focused ultrasound, these permanent blocks have the same effect as stimulation with DBS. Okay, so what about peripheral nerves and blocks under high heat? So this is something that's being used quite a bit more recently for treating painful bone metastases and basically ablating the nerves in the periosteum of those bones where those mets are. And this is an FDA-approved procedure. It's under investigation for permanent blocks in facet joints due to arthritis, and it's very similar. Again, we want to ablate those nerves. And then in the literature, we see there's nerve blocks for the treatment of spasticity. And so here they were looking at the sciatic nerves and achieving complete conduction blocks in 100% of the nerves treated. Okay, so I'd like to talk now about low heat or reversible blocks. And coming back to our friend here, it's very interesting with these treatments, if we raise the temperature to an intermediate level, about 50 degrees or so, they start to have some modulation of their symptoms. So their tremor might be modulated. If we're in the wrong spot, they might say, oh, I have tingling in my lips, and the neurosurgeon knows we have to move it a little bit. So it's very, very useful to have that reversible effect, which gives us an idea of where we are and gives us that feedback. Back into the literature, again, looking at the same group that looked at the spasticity that they found with lower exposures with the focused ultrasound, that animals recovered back to their baseline levels. Okay, in terms of stimulation, we've been doing some work with stimulation of rodent brains. As you can see here, after stimulation with focused ultrasound, they get up, they run around, they're fine. We reuse them many times. This is just showing you the very great sort of specificity that you can stimulate a very small part of the brain and get just like a single whisker moving. So moving on to the peripheral nerves. So here is some work stimulating the rat abducens nerve. This is a nerve that controls a single muscle, the lateral rectus muscle of the eye. And so you could see that the eye was moving laterally there. And so there's possible mechanisms, a number of them. We talked about heat, and many of these applications more recently with the stimulation, they have not using any heat, so they have very low intensities and low durations. So the other possible mechanisms are that there's a tissue stretch or strain and cavitation. In terms of the tissue stretch or strain, we know that at surgery you can touch the brain and have an effect just by touching. There are stretch-sensitive ion channels. And so one question is the ultrasound producing these local strains, and is that what is causing our mice to twitch? So we're looking at that. We can get pictures of what the strain looks like, or the displacement here, and then the slope of that is our strain. And so that's something, it's an area active area of investigation. In terms of the literature, there's some looking at the sciatic nerve. And here it was just an ex vivo prep. But again, they were showing that a mechanical stimulus was giving an effect. And again, hypothesizing about the stretch-sensitive ion channels. Here, some work that was combining electrical stimulation with ultrasound, and found that the ultrasound was modulating the electrical stimulation. They did this here in the vagus nerve. And so we see some change in the electrical stimulation. So I've talked about heat, and I've talked about a mechanical effect, and then the third possible mechanism is that possibly that we're creating some cavitation. So cavitation is the pulling of gases out from the tissue, and there's a theory coming out of the Technion where the theory is that we're creating a cavitation within the lipid bilayer, and then that's changing the electrical properties of the cell and creating the action potential. What's very interesting about their model is that they've been able to explain both stimulation and suppression with the different ultrasound regimes. Okay, so I'm moving very quickly. I have one last area I'd like to talk about, and that's stimulating organs or systems. And here, most of this work is using low-intensity pulsed ultrasound. There's actually a fair amount of literature showing that focused ultrasound can be used in the immune system signaling or upregulation of signaling molecules. It's a huge amount of literature for the focused ultrasound in bone and tissue healing and arteriogenesis. More recently, there's been work looking at this upregulation of signaling molecules in terms of trying to get an anti-tumor immune response. And so there's a fair amount of literature, and it's an active area of investigation. And definitely there is a lot of signaling of these molecules, and as to whether we can get the body to actually go after the tumors is still an open question. And then one more piece of work, which is really very interesting, is coming out of the NIH, out of Joe Frank's lab, where they're, again, looking at this upregulation of signaling molecules when you use a pulsed ultrasound. And their goal here is to get those signaling molecules to spatially get stem cells to home into the point of the focus. And so there's a lot of exciting work in terms of the upregulation of signaling molecules. And then just to kind of come back full circle to nerves and the tissue regeneration, there's a fair amount of literature looking at ultrasound or accelerating the regeneration of the sciatic nerve after being cut, as you can see here with ultrasound compared to not with ultrasound. And then another piece looking at sciatic nerves in rats. So just in conclusion, ultrasound has a neuromodulatory effect, both in the brain and peripheral nerves. It can be useful for blocks, both permanent and temporary or reversible. It can be used for stimulation, very active area of research, mechanical stretch versus cavitation are many of the different mechanisms that are discussed. And some of the other areas for ultrasound stimulation are bone tissue healing, arteriogenesis, immune system signaling, modulation of stem cell homing, and nerve regeneration. So hopefully in this talk, which has been very quick, I've been able to convince you that focused ultrasound is a useful way that deserves a lot more attention for stimulating peripheral activity to relieve conditions. Thank you for your attention.
Thank you. If Viviana would like to just step up for a few minutes, we have a few moments just to perhaps point some information about these talks, which not only about cross-cutting technologies, cross-cutting concepts of activation and block, of intersection of light and the genes, of electrical and mechanical stimulation. Any questions? Yes, please.
I wanted to ask about the ultrasound. One of the things we've talked about in this meeting in the past few days is specificity and targeting specific branches of nerves. Is that feasible with ultrasound? Can it be miniaturized or focused?
So there's spatial and temporal specificity where the focus is. You can control that. As to whether or not there's specificity within nerves, I don't really know the answer to that. It's possible that if what we're doing is activating stretch-sensitive ion channels, maybe there's different density in different types of nerves. So I'm not sure I can really answer that at this point.
Maybe related to that, I mean, is it possible to functionalize cells within organs or within nerves so that they are more sensitive to ultrasound?
I can comment to that. So it might be because there's, for example, a research on gas vesicles that are susceptible to ultrasound, and you can genetically encode them. So this parallels the thought process from optogenetics, where you have a protein that's light sensitive and you put light. If you have a modality that's genetically encodable and it's ultrasound sensitive, you can achieve specificity with the great benefit of non-invasiveness of the ultrasound. So you could get there.
In terms of getting light to deep tissues, have you or anyone thought about using nanocrystal technology? There are nanocrystals that excite at one wavelength and emit another wavelength, and if those can be deposited near the target tissue, one might actually be able to use infrared light through the skin to activate them. Is that something that you believe is feasible and can be incorporated into a paradigm?
That's a great concept. It recruits the upconversion property where one wavelength gets shifted to another and you get a high penetrating wavelength and then a visible wavelength out on the opsin. There was some work, unfortunately unsuccessful. We're not exactly what the community is missing on that point, but the efficiency of conversion is extremely low. So that's one problem. And then the other problem is how do you secure such centers, such conversion centers, and not have them encapsulated by the tissue. So there's a few challenges, but the thought process would be great. Go in with the penetrating modality and then emit visible light. Bioluminescence is one area where you could put emitters, local emitters, and then apply a drug, and then it emits light only when the drug is applied. So that could be recruited along the same lines.
In response to the question about focused ultrasound, from what I understand, non-invasive focused ultrasound is being used to target renal nerves for ablation therapy in humans for drug-resistant hypertension. So that is possible for ablation, not excitation, as far as I know, but it seems like that's possible.
Yes, Dr. Pauly, I want to ask you a question about your nerve regeneration data. So this is the sacral nerve with the focused ultrasound. Didn't you give us some indication that you've seen sacral nerve regeneration, or it's been published? Is there any data on anything beyond the sacral nerve, like gut nerves? We're more interested in things like the vagus nerve that may have been damaged at an operation or maybe inadvertently traumatized. Do we have any data that you could sort of vibrate, stimulate, reactivate, motivate, do anything with intensive ultrasound focus?
I wish I could answer that, but I didn't see anything in the literature on that, so I'm not sure it's there.
I think I need to add, with optogenetics, we had huge challenges at the beginning. The opsins were killing neurons. And I think early technologies, promising technologies, need to be given a chance, and lots of work is needed to control and look in all places, but this is not feasible by the few groups that do work on this modality. So,
well, thank you. I think on that note, let's move on to the next two speakers. But it is interesting how early technologies. Dr. Ranu Jung from Florida, who also had worked with the group in Cleveland, and has a background in neural engineering, and particularly in translating things from animal to human use. Over to you.
Okay, so we are shifting gears now, and in effect, it's basically kind of trying to summarize a lot of different things that have been talked wonderfully yesterday and today, and with the challenges that we face of moving things into humans. Any work that is presented from my lab was either funded by NIH extensively or some parts by DARPA. So what we're really talking about is patterns of neural activity, whether we are looking at the brain, which we always think about, but also when we are looking at the whole peripheral nervous system itself. So what we're looking at is access, and that is tapping into the endogenous activity. This is the neurosignatures, and we've been looking at technologies for that. We're talking about influence. This is the neural control problem. That is an immediate modification of activation. And then the holy grail that we all want, the plasticity, or being able to introduce neurotherapies. And so here are the questions that some of the important trends for closing the loop, and then I'm going to present some challenges that we have and just go through those. So we have neurotechnology somewhat to provide targeted neural sensing and activation, some neurotechnology with some adaptive capabilities, some neurotechnology that we are trying to increasingly integrate with the biological systems, and we are trying to get neurotechnology for more personalized precision treatment. We are dealing with a system which is a challenge. We have limited spatiotemporal sampling from the biological system. The biological system is all the time adaptive. We can only intervene with it every now and then we are trying to do a precision control of effectively the biochemical machinery. So these are the problems that we face. Before you talk about doing neurotherapies and you talk about going beyond 30 days, you got to deal with the FDA to be able to get your devices in there. So now there are many things you have to do, but even those of us who might be working in the labs, some of the things perhaps you want to pay attention to is, what's your experimental plan? Are you collecting your non-clinical test data so that it will be applicable later on? Think about your risk analysis early on, and then especially if you're designing the technology, think about your design inputs, design outputs, verification, validation, and even how the hell is it going to be manufactured and packaged, and then how you're going to use it. And there's several other things, but keep that in mind. Academic, clinical, industrial partnerships are going to be absolutely essential, and I'm not even going to talk about reimbursement. So here are the challenges. So we want to be able to get design, develop, and deliver safe, robust hybrid adaptive systems. So we've got three orders of challenges. The first order of challenges, place the neural interface at the appropriate location and keeping it there. What do we do with nanocrystals? Second order of challenges, how do we deliver meaningful stimuli and interpret the neural codes? How do we know what should be the pattern of the stimulation? Third order of challenges, dealing with variability, complexity, plasticity. So in the first part, we're talking about selective communication. Maybe we have to worry about proximity. What will be the surgical procedures? How are you going to get a stable interface? Think about chemical properties, biological effects, mechanical things, and charge density with regards to biocompatibility. Some of the things that are already being used, cuffs, and I will leave everything to Kevin to talk about cuffs. Then there's the flat interface that is reshape the nerve to get to the fascicles. And then there are intraneural approaches. This is where you are putting electrodes right through the fascicles, either perpendicular to them or in parallel to them. And one of them I'm going to talk about more is the longitudinal intrafascicular electrode. And then you have these many-shafted neuron bed of tips going into the nerves, the Utah-slant array. So extraneural, intraneural, these have all been put in humans. Then there is the regenerative things and microchannels. The question is, how much invasiveness do you want? And how much selectivity do you want? You have to make some kind of a balance between that. So here's from Fawcett's group recently that they did a closed loop for bladder control. So in this case, what they showed is that they took the dorsal root the dorsal root. They have micro-dissected that dorsal root into 100-micron rootlets. They put those micro rootlets into a microchannel device at the bottom, And then on the right, what you're seeing is on the top that if you look with the whole cuff, as you infuse the bladder, you can't really make out what's going on. And then if you and you split it in half, you start to see a pattern emerge. And then if you quarter it further, then you see that the neural activity is matching that pressure gradient even more, and then you go 8, and then you have a much better sense of it. So it's like this is the selectivity that you're getting. And in the bottom, what they show is the bladder pressure increasing, and if you had multiple channels, you will get different kinds of information from there. So this argues for being able to get more microdomain information from within the nerve trunk itself. Same thing we can do with the longitudinal intrafascicular electrodes, and this is work from my lab. And in the left, what you're seeing is that we have put into 2 fascicles these live electrodes and into the peroneal and the tibial fascicles. And when you move the foot, then what you see is that you get specific activity on one, which is flexion, and you can see with the red line going down, and then opposite is happening with the peroneal fascicle. So you get flexion and dorsiflexion. You can stimulate, and that's what's shown on the top right. And if you stimulate the 2 separate fascicles, you can actually get full ankle movement, functional movement, and you will get different, and you're looking at the muscle activity in the 2 muscles associated with it. So you can get this very microdomain sensing and microdomain stimulation, and you can control the charge you can put in. The bottom is actually intrafascicular recordings by Horch's group from a human amputee, and he was the first to have shown this ability to do that. Then you got to worry about the surgical procedures. So if you do put in these arrays, the Utah array on the right, you need to have how the hell are you going to punch that in? So you need tools for that. If you're talking with regards to the live electrodes, and these are very fine 23, 25 micron wires, which are thinner than a human hair, you've got to figure out a management system of how you're going to take them to the target tissue and put that bundling and take them to your actual target tissue itself. Then with regards to the stability of the interface, you have to anchor the electrode, and you have to think about the mechanical stresses, mechanical stress on the tissue and mechanical stress on the electrodes. Maybe you want a flexible kind of material, which is on the right that you see. It's an E-Dura used for spinal cord recordings. But what happens when you have the stretch over and over again on the leads itself there? Maybe you have different multiple sites on one lead, like you have in the commercial systems, but then these lead locations are fixed. So how do you get target to many different sites? But then perhaps you want to use a distributed intrafascicular microelectrode lead that then you can distribute to many different sites. So you have to think about all of these issues when you want to talk about targeting specific tissue, specific neurons, specific populations. Do you want to integrate your electronics with your electrode itself? That is going to pose some challenges. There may be mass, there may be heat. If you have wireless location, how far under the skin can you go? Is it going to be durable? If you think about stretchable electronics, this is fantastic. But then again, you have to worry about how you're going to access all of that. What are going to be your ribbon cables coming in and out of the body to look at that? And of course, the mechanical stresses on it. Biocompatibility, you are all quite familiar with that. Issues about systemic damage, encapsulation. This was in the brain. We know much lesser of what is going on if you went intraneural inside the peripheral nerve, and that's the other issues that you need to think about. Then with regards to this use of what kind of parameters you would use, one of the major things that you want to think about is the charge. And so in this case, what we're looking at is the reds are all bad above a particular Shannon line, and these are macro platinum electrodes. And the ones below that are the blue ones, the circles are safe based on function. All of these intrafascicular electrodes, these are studies which are preclinical and clinical, all lie within that small area. So you have got this you've got to make sure that whatever you're using is going to give you a safety region in which you can function. You need to have second-order challenges for specially distributed temporally complex mismatch between your neuron firing and organ function time scale. So you have this major difference in the two, and the context dependence. So if you deliver a meaningful stimulus, so in this case with lives, then you can change the frequency or change the amplitude, and you can get discrete and graded stimulations. And then this was done with LIFES, and recently Tyler and all did it with cuff, and you can show with frequency and amplitude you can get naturalistic stimulation. We've already seen this, the complex codes, time-scale mismatch, state content dependence that with the autonomic system. So you need to be thinking about control systems that take into account multiple inputs, multiple outputs, nonlinear mapping of your input coming in, and that will then decide how you're going to get your pulse scheduling and pulse scheduling across multiple electrode sites. You could use computational models, where Grill and Kent have done a really nice paper where they have looked at cuffs, circular cuffs versus flattened curves, and how many sites of stimulation you need to do to be able to get actual access to the different fascicles. And so that computational models could be used more and more in the design of the electrodes. And on the right is what we have done in our lab to look at what kind of recordings one might expect if you were putting live electrodes inside the nerves and you were expecting certain motor output from it. Then the final thing that you have to look at is variability across users, and this may be enhanced by impairment. We heard about this yesterday about circadian rhythms, what happens with adaptation, and then what happens, there is this variability across neural and target issues. Human factors, as you age, impairment, will they tolerate the technology? Is it easy to use? Do you need to be trained? Are you going to be working at home? Are you going to do self-dosing with this technology? And then, of course, personal preferences, what kind of activity and style you have. So when you're talking about it, then for the whole technology to be translated out to human use, you need to think about the hardware, firmware. Will you be able to make new STIM programs? Will you be able to modify them? Will you allow the user to modify them? How are you going to fit this system to this person? Because if we are talking about precision medicine using these technologies, we got to be able to fit it to the person. Calibrating the electrodes, determining the stimulation parameters, setting the channel modality, and then determining the maps and doing the studies. And then we not forget the data panel that we had yesterday, annotating your data, storing your data, and then being able to make use of it later. So I finish with closing the loop again, and in order to give safely and effectively address user needs, there is the happy news that we have technologies that are being able to do more and more and more, but we are left with these challenges that face all of us, and these are the ones that we need to discuss and figure out how we are going to address, and this is placing the neural interface at the appropriate location and keeping it there, delivering meaningful stimuli and interpreting the right neural codes, and then dealing with the variability and complexity, and especially when working with people. That's it.
Thank you very much. And finally, we'll move on to Kevin Kilgore, also a biomedical engineer based at Case Western Reserve in Cleveland. Over to you.
Okay, after that whirlwind, we will come to the last science talk. That was great, Renu. There's a lot of things there. Obviously, if we're going to take these things to human applications, and that's what we're tasked with talking about here in this session. So I'm going to, in a sense, do the second half of Renu's talk, but I decided to move up the discussion of nerve block and modulation. It came up a number of times yesterday, so I'm going to do a very brief, like 5-minute tutorial on high-frequency block, but only because I feel like it really illustrates the issues that are relevant to the SPARC program. And so I'll try to bring those out. First of all, a couple definitions, and I had to refine these even this morning. I think definitions of terms are very important. So the word block, unfortunately, is used for a whole lot of different things. What I'm going to talk about here is I'll say a temporary block, that is a block of a reversible arrest of conduction of action potentials along a neuron. And then I realized I had to define how quickly it was reversible, because even if you cut a nerve, it could regrow over the course of months. So I'm talking about reversible within seconds to minutes. Something that would be relatively difficult to do with drugs. So that's one of the advantages of of electrical block is the speed with which you could block and the speed with which you could reverse the block. And then modulation, which I'll just say a rough definition is increasing or decreasing the excitability of neurons, or a neuron or a group of neurons. I think these are particularly relevant to the SPARC program. Let's say compared to the neuromuscular system, which is what at least many of us have worked in, those in terms of the device industry. You can modulate muscle activity from 0 on up to its full activation and do it quite well. But it strikes me that in the autonomic system, there's generally a set point, and you want to go up or down from that set point. So being able to down-regulate neural activity is extremely important in this program, and I think that's not something that in the past has been a big focus, at least not of electrical technologies. So technologies available for block or modulation, kilohertz frequency alternating current, which I'll just show you briefly, direct current, infrared or thermal block, optogenetics, magnetic, and I should have added ultrasound, but my slides were already up here before Kim talked, so sorry about that, but there's another alternative. So a brief tutorial. Why do this? It illustrates some of the difficulties that we face. You know, this is really a fairly new technology, and we're applying it to not fully characterized physiology. So we have two unknowns that we're mixing together and hoping that something's going to happen, and that's bound to lead to confusion. And this is obviously in contrast to the motor neurons and skeletal muscle, which is a beautiful thing. But we've got to move beyond that if we're going to get to these applications. And also, I think it illustrates the critical need for clinicians and engineers to work directly together. If this SPARC program is going to work, these teams really have to form. The technology is not finished, and the physiology is not finished, and they're both going to have to finish it together, not separately. All right, enough of that. So quickly, in 2 seconds, 2 minutes, you will have a tutorial of high-frequency block. I'm going to go back to the somatic nervous system because it's so simple and it helps to see what is happening. So the simple issues here are that we are on the, this is in a rat, but we've tested this in all sorts of mammals, including monkeys. Stimulation of the sciatic nerve, we have our nerve block generator, and it's very simple. We stimulate here, we block the stimulation, show that the muscle relaxes as a result of the block, and what we get are things like this, and I like to put this up because it illustrates the good features and the bad features of kilohertz frequency block. So these are the twitches that you get from proximal stimulation, and those continue. We're stimulating at half a hertz the whole time. We turn the high frequency on. In this case, it happens to be a 10 kilohertz, 4 volts peak-to-peak sinusoid delivered through a cuff electrode around the nerve. When you first turn it on, the nerve fires like crazy. It fires very rapidly, basically as rapidly as it can, particularly in this first phase here. We've found out there's about 7 action potentials in this ramp up. And then in some cases it fires. It has this prolonged period of firing that in this particular case lasted about a minute before the nerve is fully, the nerve and muscle are fully relaxed. At this point you have complete block. We maintained it for 5 minutes. You turn it off and instantly the nerve goes back to conducting normally. In these conditions, the reversibility is instant. I mean, at least it's instant within less than a second. We haven't really studied exactly how quick it is. If you deliver this for an hour, the kilohertz frequency, then you will get carryover effects. The response of the nerve will not instantly recover, but it will recover. It will still recover over the course of 30 minutes to 60 minutes, roughly, again, depending on your parameters. And there's a whole space, and I don't have time to go into the specific details of that. But going back to this, so that what we have referred to as the onset response, this is obviously a disadvantage. In some applications, it's not a big deal that the nerve fires for a while, but in most applications, I think in the autonomic system, we'll find this is a major impediment, and we have been working on ways to get around that, but I'm not going to really go into those. There was a question about whether you could do graded block with kilohertz frequency, and you can't. You can grade it extremely well. This just shows, as a function of sine wave amplitude, the percentage of, in this particular case, it was muscle force, the percentage of muscle force that was blocked. So you can do a nice graded contract or a block of neural activity, and this is a big advantage. You don't have to block the whole nerve; you can block parts of it. The question is, what fibers are blocked first? What fibers are blocked last? Unfortunately, it's just like electrical stimulation. You block the largest fibers at the lowest amplitude. You block the smallest fibers at the highest amplitude. And I wish it were not so, but it is. So what you first block, this is in a sensory nerve. This was in cat. I should have put this out. This is cat sural nerve. So the beta fibers are blocked at about 4 volts peak to peak. And as you go higher, you progressively block larger fibers. We haven't shown, but Rob Butera at Georgia Tech has shown a block of C fibers. I think there's still some work there to really verify that works in mammals. So just quickly, some of the effects here. The block actually occurs within about 10 milliseconds, although in most cases the onset activity kind of obscures when the block begins. It's quickly reversible, strongly dependent on amplitude. You have to get high enough. I'll show you a map in a minute. There's a lot of unexplored territory here. The response is complex to these high-frequency waveforms. These are way out of the physiological range, so it's not surprising that the nerve response is more complex than when we're down in a typical range of 10 hertz or so. And almost all the work to date is in myelinated nerves. And so when we talk about the SPARC program, we're going to need to know what happens in unmyelinated fibers and these smaller fibers. So just a quick contrast, and then I'll be done talking about kilohertz frequency block. So this is at 20 hertz or 5 hertz, whatever you want to do. You get one-to-one activation. There's some threshold. It doesn't matter how long you deliver it. The nerve fires one-to-one with stimulation. When you go to kilohertz frequency block, you get that same map looks like this. So there are all sorts of different regions in here. So here's the block region, which is the data that I showed you, but there's also a region in which you get a high degree of activation that continues for a long time until something fatigues. There's actually a subblock region where the membrane is being rapidly alternating between depolarization or hyperpolarization, but it's not doing anything. Action potentials can travel through fine. We verified experimentally that this occurs. It's a really weird state of the muscle. And then there are states that change depending on how long you've delivered the kilohertz frequency. So the point is, if you don't know where you are in this space, you really don't know what you're doing to the nerve. So I don't want to pick on Interomedics because they did a lot of work in getting things together, and I have a lot of respect for anyone who's able to take things all the way to market. So, but the point is, you don't really know exactly where that application is working. So we're not sure whether it's activating, blocking, whether maybe there's some carryover effects based on the time of delivery, et cetera. Okay, so I have to wrap up. There are some things that we don't know. I tried to identify areas of work that I think should be or could be a part of the SPARC program. And this isn't just for kilohertz frequency block. This really goes for any of these modalities. But in particular, kilohertz frequency, the generator itself needs to be characterized. It turns out that it's not that easy to generate a current-controlled, high-frequency wave, 20 milliamp, 30 kilohertz waveform and deliver it through an electrode to the nerve. It's not that simple. It's not as simple as you might think. We have not found any device that delivers what it says it delivers off the shelf. So don't just pull off some grass stimulator and voltage to current converter and expect it's going to work. It won't. You have to know where you're at, whether it's activated, blocked, or whether the nerve is cooked, which is a good way to block, by the way, but probably not what you were intending to do. It really requires a methodical approach. I really like what Brandon and Marion did, starting with nerves, a nerve sitting outside the body, stimulate on one end, record the action potential on the other, first verify that and then take a progressive approach before you start sticking a high-frequency electrode in the middle of a complex system where you don't know what it's doing and you don't know really what the outcomes are of your whole system. All right, I've said that enough. DC block, we can talk if you're really interested. I think this is better than high-frequency block, but anyway, we'll go on to that. But you can do it. You can do it repeatedly, at least in your acute studies. You ought to think about DC blocks. Get an electrode, coat it with some cotton and saline. You can do DC block for a couple hours that way without damaging the nerve. We think you can actually do it chronically without damaging the nerve, and we're doing some work on that. So relevant to Spark in terms of nerve block and neuromodulation, areas for research and development, getting a consistent and repeatable block, chronic safety, any safety data that's there from the products that are out on the market is not published. Unfortunately, it's not available to us, so we're going to have to do this probably as part of the program, small fiber block and targeted block of small fibers. Am I at 0 minutes yet? Oh, okay then. Well, all right, cuff electrodes. We'll skip all of those wonderful things. I will say one last thing. So we haven't talked a lot in this meeting about sensors and sensor modalities, and that obviously is a critical part of the technology in the Spark program. So whatever is reflected at NIH, I think it really needs to include that as well. It's just none of us have had time to get to that point. We've all been talking about activation. I think that's all. Okay, I am done.
Thank you, Kevin. We just have 5 minutes for discussion. If all 4 speakers could come up to be available for any points, and we have the first question at the microphone.
Thank you, Kevin. Is it feasible to have an electrode that can provide a stimulating activation of the nerve fibers, as well as use that same electrode and pulse generating equipment to block? So for micturition, we'd like to keep the sphincter closed, and so stimulation of the pudendal nerve would be great. When we wanted to void, block that pudendal nerve. Could that same equipment be used in a setting? Is it feasible?
Yeah. So for the high frequency block, we're using nerve cuff electrodes. It's a big benefit to have good control over how the current's flowing in and out of the nerve. So nerve cuff electrodes work great, but it's the same nerve cuff electrodes you'd use for stimulation. So you could do the same through the same device.
Thank you.
We have 3 speakers lined up the microphone, so please, would speakers and responders be brief?
Another hurdle for nerve block, I'm wondering, is knowing that you're actually blocking the nerve in a patient. So if you're trying to treat obesity or whatever, you can have contact, you can have the output that you can measure, but how do you know you're actually blocking conduction without recording from the nerve or something like that? Isn't that an issue?
Okay, so I think that's actually a very important point, and I'll try to be brief, Graham. But I think we ought to consider, when we get to human studies, of including in the devices some sensors that are actually specific to identifying what the technology is doing and may not necessarily contribute to the function provided to the patient. So, for example, recording nerve activity upstream and downstream from a block or activating electrode, and having that information from humans would be a huge benefit to us. So I don't know if there's any issues related to adding technology into a device that just provides data back to us. But I think we should seriously consider that, because you're right. You don't know if it's blocking, and you really need that information. If you're going to progress the technology at all and progress the field at all, you need that information from humans. Obviously, you can get it from animals, but it would be great to have that information.
Bob Hoyker at Children's Hospital of Philadelphia. So I wanted to just take a moment, since we have so much accumulated wisdom in the room here, to make sure that we think about pediatric applications as well as adult applications. And I'd be very interested, because there's actually nothing that I heard about pediatrics. I'd be very interested in thoughts about how devices need to be designed differently. What do we actually need to know about the pediatric nervous system to be able to take advantage of these technologies? How do we integrate pediatrics into this really new and exciting area of nerve modulation?
Thank you. I think that's a large topic and a good one. And there's been some work in the neural prosthesis world on what to do as a child grows. So perhaps that's a question we can take into consideration for the program as a whole.
Thank you.
Yeah.
We've just talked about invasive technology. Any comments on non-invasive neuromodulation techniques?
No. No, actually, those were some of the slides I had to skip over. But like ultrasound, of course, is certainly one. But one of the things that wasn't mentioned yet is magnetic stimulation. So obviously, it's used for transcranial magnetic stimulation, but also can it be used in the periphery? Even surface electrodes. I hear that there's a lot of autonomic fibers in the skin, so you can certainly access them with surface electrodes. So, I think it should not be ignored, some of the surface, external technologies.
Hi, well, thank you, everybody. I think this is a session on tools and the toolkits that are displayed by the four speakers bears a lot more examination, obviously, but we should bear in mind that these are tools, they're means to an end, they're not the end. Even the mapping is not the end, that our mapping should be driving towards medical interventions. Thank you.
So the next two talks, we've invited two speakers from our sister programs, should we say? I feel like we've been doing a triangle tour of talks. So Doug Weber from DARPA will be talking about the Electrics program.
Thanks, Grace, and thanks to everyone else on the Spark team. And thanks to the speakers. The quality of the presentations and discussion over the last 2 days has really been excellent, so I appreciate that. So I come from DARPA, which is a sister or brother organization to NIH, certainly a federal agency, federal funding agency that has a significant interest in biological technologies, so much so that just in April of last year, we created a whole new office within the agency to focus specifically on biological technologies. And a major portion of our portfolio in BTO is really on neurotechnologies. Much of that has been to date in the sensory motor prosthetics domain, but with our Electrics program and some of our other brain stimulation programs, we're looking at other opportunities to create technology that integrates with the nervous system to restore or enhance functions, particularly the ones that have been lost by injury. So Electrics is a brand new program. We just closed the call for proposals yesterday at 4 o'clock, so sorry if anyone in the room hasn't heard of it and wants to submit a proposal. The good news is you can talk to me now, and so I can maybe provide some guidance. But our vision for Electrics is really very similar to the ones we've been talking about. We really want to, like everyone in the room, advance the field of neuromodulation and use the nervous system to provide kind of new access to treatments for, in our case, things like inflammatory diseases, mental health disorders, perhaps other DoD-relevant conditions. But in all cases, what we're doing is we're looking to the nervous system to provide that new ability to regulate physiological functions. So a lot of similarities between Electrics, Spark, GSK, and others that are working in this space. We're going to try to do some things that are distinctly DARPA in the sense that we're going to set some very lofty technology goals, some very ambitious science discovery goals. But across the board, our goal is ultimately to identify and understand circuits that we can then interface with to monitor and control neurovisceral functions. And we really want to understand not only the function of those circuits sort of natively, but understand what happens when we place a device into the nervous system to try to access those functions. And we want to take a very global look at this so we understand not just what's happening on the target that we're trying to modulate, but what's happening outside that target zone as well. So from a science standpoint, we want to learn more about the physiology. From a technology standpoint, we want to use that knowledge of the physiology to create new devices that provide improved ability to precisely target cell types and numbers of cells. And we want to move that technology in a direction where it can be deployed through minimally invasive means. Okay, one of the barriers to adoption with existing neuromodulation devices is the surgery that's required to adopt those devices. And in many cases, those devices are seen as a last resort. It's a lot easier to go down to the pharmacy and get a drug than it is to have surgery and implant a device. So clearly, there's room to make those devices function better, but I think there's also opportunity to make them easier to use. And then one other thing that's been touched upon, and Kevin mentioned it in his closing comments, we really need to provide feedback for these devices. In many cases, we have no real way to monitor what's happening when we turn the device on or adjust its parameters. And so, for a motor prosthesis, the feedback is obvious. Does the muscle twitch? Do you generate enough force? But if the spleen is your target, it's much less clear how you know when, in fact, you're getting the physiological response that you want. So I think I agree with Kevin and others that we need to do more to develop sensors that can be used to close the loop. So that's sort of our overall goal. I probably used my 7.5 minutes on that one slide. Maybe what I'll do is just move quickly through the way we've structured the program. This is sort of our toolkits and parts list slide. So we've divided the program into two distinct areas. Technology is one of those areas. That's our toolkits, I suppose. And physiology is the other area. And I think our objectives in each of those areas are clear. We want to understand the physiology and create technology that can work with it effectively. About scheduling, so we divided the program. It's a 4-year program right now, divided into 2 phases, with phase 1 focusing mainly on establishing proof of concept, so identifying actionable targets in the nervous system and characterizing the physiological response to neuromodulation. And then on the technology side, we are exploring hopefully we'll be exploring very novel devices for monitoring and modulating neural activity, as well as measuring biomolecules in the body. And we want to really pursue unconventional approaches. I apologize to all the electrodes people in the room. We specifically excluded electrodes in here because we want to, electrodes still serve us very well, but I think the discussion over the last 2 days has really highlighted some of the longstanding limitations that we face with purely electrode technologies. So there's lots of other people that will continue to support electrode development in electrics. We're going to look for alternatives. And then phase 2 of the program will hopefully build on some of the proofs of concepts that have been demonstrated in phase 1, moving towards closed-loop neuromodulation of the target systems and establishing long-term chronic viability of our interface capabilities. Timeline, so as I mentioned, the call for proposals, the so-called BAA, closed yesterday, and my team and I will spend the next month or so reviewing proposals, and we hope to kick off the program late summer, early fall. As we're rolling this out, we plan to continue working closely with the NIH to make sure we're synergizing our efforts appropriately. So it's possible that in the future, when we hold meetings to bring our teams together, we'll invite the NIH folks and their performers or their investigators to partner with us. And then lastly, this is clearly a large space. It's going to take a community of not just scientists, but industry and government stakeholders to come together to advance the science, advance the technology, and then make that important step of moving it out into the commercial market. And I really want to highlight the supportive role that the FDA continues to play in many of the DARPA programs in this space, and I'm sure they help many others as well. But we really see the FDA as a partner in all of these programs. We work with scientists and evaluators at the FDA, and I think it's important to recognize that they want many of the same things we do. So I appreciate that. And with that, I guess I'll turn it over to Roy, and we'll take questions at the end.
Thanks, Doug. So I'm delighted to introduce Dr. Ray Katzo, who is the manager of the Open Innovation Challenge and External Partnerships Program at GlaxoSmithKline.
Thank you very much, Grace, for the invitation to speak at the meeting. Good morning, everyone, and greetings to those of you on the webcast. I think over the last day and a half, we are all aligned on the fact that in spite of our best efforts across the healthcare sector, including pharmaceutical companies like ours, there is still huge unmet patient need that we need to address. And we feel that neuromodulation does offer us an opportunity to potentially specifically intervene in those settings and help address that unmet need. Now, clearly, if we're going to do that, there need to be a number of foundational blocks that need to be in place. I'll echo exactly what Kevin said about, making sure that we got some clear functional and anatomical mapping of circuits of interest. I think over the last day and a half, we've talked about the importance of really considering how we recruit patients into the clinical trials, how we design those clinical trials, whether we're actually optimizing the treatment course in terms of the stimulation and recording that we're doing. And then, of course, how we understand how the system adapts over time. Now, clearly, that is a lot to do, and it's not something that a single company like GSK can do on its own. So the importance of partnership, which Doug ended up on his slide, is one that we passionately share. I think this will take collaboration not only for industry partners like ourselves, funding agencies like DARPA and NIH, but also bringing on board academic groups, surgeons, patients themselves, because we'll need to convince them to adopt this. If we are really going to realize the potential of neuromodulation devices, not just being treatments of last option, but potentially being first-line treatment and competing with vaccines and small molecules. That is the long-term vision of where we want to go. So I think as a group, my challenge to all of us is if we're sitting in this room in 10 years' time discussing the same issues and problems, then we would have failed miserable. So what are we doing as a company within GSK? I just want to cover 3 specific aspects that we're focusing on at the moment, and then I'll end on my second slide with where there are sort of partnering options moving forward. Now, yesterday we had quite a lot of discussions on the Vega system, and I believe Kip thinks that if you interact with the Vega system, you even have an opportunity to solve world peace, which is quite interesting. Now, what we have done is to try and explore the other networks that lie beyond the vagus nerve. So at the moment, we're trying to rapidly test some of these hypotheses across our disease biology, which includes sort of 21 collaborative projects across the globe. And over the last day and a half, you probably would have had the opportunity to hear some of the PIs who are contributing to that effort, the likes of Brendan, Jay, and Kevin. And we're looking at 16 different circuits across 24 different circuits across 16 chronic diseases. In terms of technology, we are looking at 13 different nerve interfaces, and I think we had an eloquent talk earlier on today about the different nerve interface approaches that exist. We'll be trying to look to see whether we can build something that gives us the high resolution that will be required to help us understand some of these discrete circuits. Through our venture capital fund, we're trying to nurture some of these early manifestations of bioelectronic medicines, as well as some of the key platforms that will play a central part in the future of these medicines. So things like surgical techniques that are less invasive, that are likely to drive uptake of these treatment modalities, material science, the energy aspects that we will need to address. And to date, we've made 3 investments across that portfolio. And then, of course, we're committed to delivering more tools that will be available across the academic sector. And we're doing this primarily through our Innovation Challenge, which really stems from a meeting that we held in New York in 2013, where we invited 140 folks from across our partner funding agencies, academics, etc., who identified the need of having a chronic modulation tool available for us to interrogate, circuits of interest as being a key challenge that needs to be addressed. And as a result of that, we have put forward a $1 million award for the first team that develops this solution, but we've also injected an additional $5 million to make sure that we support those teams throughout that journey. Now, as of last year in December, we announced 10 teams across 4 continents that are each being funded to the tune of $200,000 over the next 7 months that will be looking to de-risk certain aspects of building this device. And by the middle of the year, we then intend to inject an additional $1 million each for up to 3 teams to then try and build these systems. Now, clearly, these systems are going to be very different from treatment ones, and I think that echoes the comment that Viviana made earlier on. But I think if we can build these miniaturized platforms, it then gives us foundational blocks to then move into preclinical as well as ultimately clinical platforms that will be required in the long term. So what are we going to do once we got all these building blocks in place? Well, clearly, one of the things that we want to do is to link circuits to the effect on disease. And we can tell you that we are committed long-term to being a partner that will be involved in chronic neuromodulation experiments once there is sufficient reason to believe that we should go after certain circuits. We'll be looking to develop and deploy the high-resolution sort of peripheral nerve interfaces that will stem from our exploratory portfolio, as well as the research platforms that are likely to come out of our Innovation Challenge. And of course, echoing the statement I made about partnership, we will seek to complement the efforts that Electrics and Spark programs are driving to make sure that there is synergy across the space. And we also expedite the ultimate treatment modalities that we all desire. We intend to be a path to the clinic and patients. I think yesterday we had quite a few talks where there are still situations where there is a disconnect between what we find out in our disease models and what happens in the clinic. Now, we clearly want to be part of a solution in terms of addressing that. And that is something that we hope is going to happen in the short term and long term. And that's where I'd like to end up. So finally, just to wrap up, I think this is a vision that's worth pursuing. I think having meetings like the ones we're having today that bring together all these different parties is to be applauded. So I thank the Spark team and the organizers for giving us the opportunity to meet up over the last day and a half and share our collective vision and path as to how we'll ultimately get where we need to. Thank you.
Thank you very much, Roy. So I'll bring up the final session chairs, Dr. DeGroat and Dr. Kilgore, to have our opportunity session.
Oh, we can leave that. Let's go back to the other one first. Yeah, let's want to go back first. Yeah.
He's first. That's first.
Yeah, right. Okay. So these were some of the marching orders that Kevin and I received. And this reiterates the goals of the conference. So talked about building detailed and integrative functional anatomical peripheral neural circuit maps in multiple organs in humans. My response to the use of peripheral, and we talked with Grace and Jill about this before, is that I think neuromodulation depends on information not only about peripheral neurocircuits, but also about central neural pathways. So I think that we should, in the future, be expanding the SPARC program to include at least something about central reflex mechanisms. So then what information do we need in order to build the maps that guide specificity and efficacy of neuromodulation approaches? I think we did talk a lot about the parameters of the peripheral nervous system and anatomy, fiber sizes, conduction velocities, neurotransmitters, and others. I think we did a pretty good job of this at the conference. There are lots of specific places, sites, examples that need to have further information. And clearly we need to understand the similarities and differences between humans and animals. And that came up many times at the conference. Does something actually that you identify in animals apply at all to humans? I think Nico Rykov certainly brought that up with examples of lower urinary tract modulation. What parameters of organs need to be understood and what assays developed in order to assess nerve function and eventually therapeutic efficacy? I would say we probably need to understand both physiological normal functions as well as abnormal functions. Ideally, I'm a pharmacologist, but when we talk about treatment of disease or symptoms, we ideally would like to target the abnormal, eliminate them, and restore normal function. So that's the ideal. But in many cases, especially with the autonomic system, we really don't know what to target. We haven't identified the pathophysiology. And I think that's a big gap in understanding how neuromodulation works, because clearly it's going to target, at least in part, abnormal functions. And so in the urinary tract, for example, we have idiopathic overactive bladder. We have idiopathic urinary retention. Idiopathic, we don't know really what the cause is. But on the other hand, we also have to know about the normal physiology. For example, you can treat hypotension without knowing exactly the cause of hypotension by simply giving agents that can increase cardiac output or increase peripheral vascular resistance. And on the in acute phase, you can bring blood pressure back up. Or you can simply tilt the person's legs up so that you increase venous return. So we really need to know both normal and abnormal function. And then how can animal and human data be leveraged to generate useful human maps? Yeah, I think this is where technology comes into play. As we get new techniques and devices in our hands, often we start to learn new things about the physiology of the heart, for example, as we heard yesterday. Beautiful story about the cardiac ganglia and the integration between afferent and efferents from the sympathetic and parasympathetic pathways to the heart. So imaging, clarifying tissue, as we heard this morning, so that we can get a better indication of the innervation patterns within the organs, et cetera, is really going to help us to make the transition between animals and humans. So as I was sitting here this morning, I was jotting down a few things, like my own biases. What interests me, of course, is what I remember from the conference. And there's really been a lot of information that's been transferred between all of us and to the people that are listening or looking on the web. And we've all been educated, and we will have some brain plasticity as a result of being here at this conference. We're different people than when we came in, and we're going to think about the autonomic nervous system in a different way than we did before this, and hopefully that will translate into new approaches, new ideas that will be explored back in our labs and in our clinic. So it does seem worthwhile to briefly summarize some of the highlights of the workshop. And Kevin will deal with technology, and I'll deal with the biology. The one thing that struck me was that the major focus of our meeting was really the vagus nerve and the organs that are innervated by the vagus nerve. I counted about 13 talks that dealt with biology, and 10 of them dealt with the vagus nerve. And maybe that reflects its broad distribution in the body, how large it is, maybe how accessible it is, how many of the important functions in the body are controlled by the vagus nerve. We had beautiful pictures of the morphology of the gut afferents from Terry Pally and Ellen Raybould, the conduction velocity, the fiber sizes, the physiological properties, the sensitivity to various stimuli. And then we heard about unusual properties, how the vagus can control the immune system and in the spleen. And so we've had considerable coverage of the peripheral nervous system, the afferent and the efferent pathways in the vagus nerve. But what struck me also is, with exception of maybe the cardiovascular lecture or the cardiac lecture, we really didn't touch upon at all what happens in the brain when those afferents from the vagus go in and trigger something coming back out again. And again, I would emphasize that is an important component of this. Dr. Tracy told us about how the vagus can pick up this information about fever and various stimuli, and then how it can come down and affect the immune system. But what about up there in the brain stem? And I was going to ask you, if there was time, have you done studies using transneuronal virus tracing techniques to see what types of circuitry are present in the brain stem and maybe higher up, which are controlling the spleen and the pathway to the spleen? So I think that's a real gap in what we covered here and in our general knowledge. So neuroplasticity in vagal afferents was discussed by Helen Raybould, and how that can change the expression of various genes. And certainly plasticity is important for understanding pathophysiology, because I think we all would agree that disease can change the function of the autonomic nervous system, both the afferents and the efferent system, as well as the organs. And I think this is something that emerged here. We discussed how non-neural cells can impact the nervous system. We heard from Helen about the enteroendocrine cells, and you may remember all the way back to the first day I talked about bladder urothelial cells and how they can release chemicals and talk to the afferents and change afferent properties and sensitize them. And I'm sure there are many other examples that you have here that have not been brought up where neural, non-neural interactions are an important component of this visceral nervous system physiology. So I think we need to know a lot more about neuroplasticity and non-neural neural interactions within the viscera. And then it came up this morning, I think maybe Carl Thur or somebody mentioned it, that maybe electrical stimulation itself induces plasticity. Electrical stimulation of the nerves, that is the afferent nerves or the efferent nerves, or as was mentioned in the cardiac ganglia, can change the properties of synaptic transmission. It can actually change gene expression and change the insertion of various ion channels and change the sensitivity or excitability, the response to stimulation, so that it's possible that neuromodulation might get better or worse as a result of plasticity. Very often the body is smart and it tries to actually counteract what we want to do to it. And that is a form of plasticity or adaptation. And so you might find in certain cases that the stimulation is less effective as you go on, just as the response to a chemical neurotransmitter can be downregulated when it's applied in an abnormal way or in high concentrations. That's called desensitization. The other thing that I was struck by in the cardiac lecture, let's see, are we running out of time here? Yes, move quickly through the summary part. Okay, so I've gotten my message. The summary part apparently is what were submitted by Grace.
The session chairs. Submitted by the session chairs.
Yeah, that's what I'm saying. That's the session chairs, right? Okay, so we don't want de Groot to put all of his biases into this. Okay. You want me to go back to the first one? Okay. So was this biology or was this technology? I wasn't. Okay. All right. So I guess it's my opportunity to do some reading. So so the summary from the session chair, then summarized further by Grace and She wanted us to give you her work and put it up on the screen here. Okay, so we need better characterizations and definitions of what electrodes are actually doing. Okay, with which fibers and mixed nerves. Yeah, I guess that is indeed true. We need to know how they stimulate the nerves and what nerves are being stimulated, and eventually what that stimulation does. And I guess in my view, a lot of stimulation releases chemical agents to trigger various types of organ responses or neuronal responses. Then we can make this an open discussion if we have a little bit of time. Let's get some feedback from this. Is there any other thing that electrical stimulation could do besides changing gene expression in the nerves themselves, or by changing the impact of the nerves on their subsequent target?
Yeah, or perhaps you could just run through what the major points were as kind of seeding the discussion, and then we can open it up to everyone to add.
Good. So you can see that people have had a lot of experience in running these meetings know how to get through the final session. Okay. Need some discussion of the disjunctive results that often surface when open trials in Europe versus randomized clinical trials in the United States, I guess, are compared, and need to find paths to get from phenomenology to mechanism. I would definitely agree with the last one. We need to understand mechanisms of action. Comments, additions, criticisms of those. We talk about terminology. Now, again, throwing my bias in. We've heard a lot about sensory afferents. What are sensory afferents? I thought afferent fibers are activated by stimuli and essentially are sensory in nature. But what are sensory afferents? I heard that about 5 or 6 times this morning.
In fact, they should be called visceral afferents.
They could be visceral afferents? Yeah, that seems like it would be a better term for me, versus somatic afferents. You could actually say that there are afferents which are reflexogenic, purely reflexogenic. That is, when they're activated, they trigger, they don't impinge upon sensation. They actually trigger some organ response or response of neurons at some place, other place in the nervous system.
But it will be the origin of their, where they come from.
Yeah. They could be bladder afferents or colon afferents, yeah. But sensory afferents makes no sense to me. So somebody wanted to dispute that or debate that. Not a big question, but we had a discussion the other day about terminology. All right, we can move on to the next one then. Okay. So this one was the lower urinary tract. And this from Helen Raybould really summarized what we talked about, not necessarily opportunities for the future. Bladder has 2 main functions, storage and elimination. This is binary, may be different from other visceral organs where function is graded. In that sense, it might be then interesting to know how the neural circuits are organized to generate that kind of function. If you have a system of an autonomic nervous system that has graded responses, that is constantly turned on and, I say, generating propulsive movements in the intestine or maintaining blood pressure between a very narrow limit, and then another system that works like a switch, it'd be nice to know what the nervous system actually, how it creates those kinds of very dramatically different controls. So many of the pathways for reflex function are quite well described. All the changes in circuits that occur in pathophysiological, such as overactive bladder, are not known and may involve altered activity in one or more of the components of the system. Yeah, and so I think here again, we have to know pathophysiology. Let me just add a little bit to that. We know that from the studies that were described for the respiratory tract, that afferents, especially the ones in the lung, can be activated by capsaicin, by acid, by all kinds of insults. And we have the same in all of the other organs as well. Some of these are mechanosensitive, some of these afferents are chemosensitive. But we should also remember that neurotrophic factors it can produce changes in afferent sensitivity or induce sensitization, which are more delayed in onset and more prolonged in effect. And so one of the issues that was brought up in organizing the conference was biomarkers. And I didn't have time to talk about it the other day, but people are measuring biomarkers, in quotes, in the urine. And neurotrophic factors like NGF and BDNF are being measured in the urine and shown that the levels are correlated somewhat with the type of dysfunction, the magnitude of the dysfunction, and actually to reflect treatment. And so I presume in the respiratory tract, Brendan Canning may comment on this, that you can also remove fluid from the respiratory tract and measure biomarkers that might be released there as well. So I think that we would like to know how these trophic factors and these slowly reacting substances actually work, what's their mechanism, how they are released, and then how they are removed from the system, and how treatment does impact their levels. The other thing that I forgot to mention just a little bit earlier, and that is what about the release of various chemicals or transmitters from non-neural cells, which are talking with neural cells? That is epithelium or neuroendocrine cells. How do transmitters get out of these cells? Transmitters released from mast cells, or T cells, which we heard from Kevin Tracy. We think that transmitters, the traditional transmitters, are released by exocytosis. That is, they're stored in vesicles. Those vesicles transport to the membrane and then get dumped out. What about the release from T cells? We don't know. The release from epithelial cells in the bladder probably involve multiple mechanisms. Acetylcholine seems to be released by an organic cation transporter. ATP can be released in part by exocytosis, blocked by botulinum toxin, can be released by channels called pannexins. All right, you heard that. And sacral neuromodulation is being used and can promote both storage and elimination. And how does this occur? I think maybe I told you a little bit how we think it might occur. What neural structures are being stimulated? I think we need to know more about the somatic afferents, which are used to trigger neuromodulation. Is the site of action peripheral or central? Mainly central, with a lot of different modulations, but in some cases also peripheral. And we know something about the neurotransmitters. Thank you, Helen, for those comments. Now we go to the cardiac sympathetic afferents and the impact of sympathetic excitation in cardiovascular diseases such as heart failure and hypertension. Strategies that need to be undertaken are selective ablation with RTX. I thought that was an interesting story this morning about RTX, also in the gut, and by Jay Pashrika, and how that can impact cardiac function and the afferent system. Cardiac effects on afferent neuropeptide release. Selective ablation in patients may be data from patients treated with chronic or for chronic pain. Yeah, I think the cardiac afferents and sympathetics are really interesting. And we were talking yesterday privately about whether there might be or branching afferents where the cardiac afferents may go to the lung as well as to the heart, and that this may contribute to some of the similarities in cardiac and lung or respiratory function and blood pressure changes, et cetera. So we need to know more about these branching afferents. How do you take technology developed for mapping in animal studies to that for mapping in human subjects? That is a big question. And do you do mapping in donor tissue? Can you extend beyond brain imaging or spinal cord imaging or organ imaging? I don't know. I don't really have an answer to that one. What approach could you take to select parameters for stimulation? How important is it going to be to understand CNS functions along with PNS functions? I would definitely vote for that one. I think they're tied together. The vagus nerve innervates multiple organs. That's true. How important will it be for us to understand organ-organ interactions? Very important. This has clearly emerged at this workshop. Any comments about these? Any additions? I think these are all true related, and important.
Well, I'd
Yes.
like to make just one addition. There's been to me, a glaring lack of comment about short and long-term changes in transcription, leading to changes in gene expression. This is really important at nicotinic acetylcholine synapses, for instance. I think if neuromodulation has an effect on both short and long-term changes in gene expression that can affect chemical coding and synaptic function, that we really need to have a better understanding of how this goes on the structure-function maps that we've been talking about.
Yeah, no, I think no question about it. The long-term changes, which probably involve really interesting mechanisms like trophic factors and alterations in intracellular signaling pathways, et cetera, important issue. In the bladder, for example, NGF goes up in the obstructed bladder and spinal cord injured bladder. The afferents are sensitized. If you use an NGF antibody, you can prevent that or reverse that. So, and this is being done in a lot of different systems. So, important issue.
I'd just like to make a comment again, being a pharmacologist. When you're trying to dissect out these central pathways, and you're obviously stimulating fibers that can inhibit what you're trying to achieve or mediate what you're trying to achieve. And I think pharmacology is a great tool to understand what is actually going on. So, and even more importantly, clinically, you may find that, yes, you can have a low stimulation intensity, you give a low amount of a drug, and you can actually facilitate. So if you think serotonin's involved, you use a serotonin reuptake inhibitor, and all of a sudden you've multiplied the effects that you're going to get. And conversely, if you think when you're stimulating the pudendal nerve, some excite the bladder, some inhibit the bladder. Well, if you were able to give a drug that would damp down a bit of the inhibition, the effect you don't want, I think there's both a basic science benefit from that as well as a potential clinical benefit, because I don't think electrical stimulation on its own is going to give you the specificity that, it's not going to be a panacea. So again, I think we have to look at all of these tools, use them in combination to really get a good effect in the patient.
Yeah, thank you. Glad you got that in. That was a part of my monologue that Grace and Jill appropriately interrupted. But I think that's one approach to go from animals to humans. That is, you identify a chemical message in a mechanism in an animal, and now you use a drug that is safe to use in humans, and you can manipulate that chemical message, and then see whether similar changes occur. For example, in Dr. Tracy's experiments, he has acetylcholine implicated in this pathway for immunization. What happens if you enhance the responses to acetylcholine by preventing its breakdown using an anticholinesterase agent? Does this enhance that response? Yes.
So I'd like to second that. Both of the two comments we heard about the importance of looking at transcription factors, the importance of using pharmacological tools, but I think we should leave on the table as a very important point, we also want to embrace the use of molecular genetic mechanisms. This is now the coin of the realm in mechanistic science approaches in neuroscience, immunology, physiology, and on the list. And I think we should have it, sort of on the record that we want to embrace the current advances in molecular biology to nail these mechanisms that guide the development of our technology.
Mm-hmm. Jay?
On that comment right here, it's a confusing factor, though, too, in terms of many of the clinical trials on devices are in people who are on drugs, and the drugs aren't working, and then we look at the result of that trial, as opposed to looking at just that intervention itself without the drugs. So I think most of the trials I'm aware of in hypertension world, they're on 5, 6, 7 drugs, and then you look at what the treatment does. So that also confuses the issue.
But some of those drugs are really not working very well because they're probably not targeting the pathophysiology, and if there's effective neuromodulation that's been identified in animals, that might be leading us to a new target for the treatment of pathology.
Or some of the drugs may ramp up, for example, the sympathetic nervous system activity that then you target. I'm just saying that's a confusing factor that we have to keep in mind.
That's true. Yep. Dr. Keast.
So I just like to put a bit in for sex differences here, because there's some very obvious ways that might be important. I guess in sacral nerves, etc., we might think that the parameters that you would need to selectively activate, say, lower urinary tract pathways might be different because you've got contributing populations of nerves that are quite different. The reproductive innervation in one versus the other may have different populations that will impact on how to separate those out. But there may well be relevant points in other parts of the nervous system too, whether it's the immune control or others, where we might want to consider separately doing those measurements and trying to distinguish the differences. That must be my earrings kicking in there. Sorry about that. Sorry about that. So I don't know if you want to bear that in mind, particularly for some of your particular neuromodulation studies.
I definitely think hormonal neural interactions is an important issue, and I think people are getting around to studying that in more detail now. And we also heard about pediatrics this morning and development. And I think some of the developmental changes that you see in some systems may actually give you some insight into pathology. In the situations that we study, pathology actually causes reemergence of early neonatal forms of reflexes. And electrical stimulation may actually affect the development of pathology and that whole process of the change from reversion from an adult back to a neonatal form. So,
I mean, your typical adult rat experiment that I see in the literature is a 6-week-old rat, which is not actually a mature adult. It's sort of on the way there. And so, again, that might affect some of the ways in which we want to manipulate those nerves. If I could add one other thing, which is a bit more lower urinary tract specific, but also applies to other organs perhaps, is that by simply looking at an organ as a uniform tissue, we might be missing important things. So in the bladder, for example, the sensory innervation is highly focused. The suburothelial innervation is highly focused to the bladder base. And perhaps by learning more about the signals coming back from different areas or the ways we can respond to those signals in a feedback loop might be very important as well, rather than think of it as a uniform target.
Yep, I agree.
I was just going to add to that yesterday we also had just a mention of aging. You can repeat.
She is interested in the issue of aging and how aging may impact, neural control as well as organ function, and I certainly can attest to the problems of aging, and so I think it should be studied more. Yes, Jay.
I'd like to toss out something that's probably provocative. So I think today and probably yesterday, which I wasn't there for, and I've advocated this myself in my talk, is let's deconstruct this peripheral nervous system as it applies to visceral organs. And let's try to tease out the anatomical, physiological maps so that we can manipulate them better. But if you pause and think about it, this is the only system where so many complex nerves have been thrown together into one bundle. And even so, you have a vagus, you have the sympathetics, you have the spinal afferents, then you have all these ganglia, which we still don't fully understand. The celiac ganglia, the prevertebral ganglia, the paravertebral ganglia, and all the others. So everybody's communicating, talking to each other all the time. And then when you go to the organs, this dichotomous, trichotomous innervation exists. So what is the, teleologically, is nature trying to just be anatomically frugal? So say, let's just put it in one bundle and not worry about it. Or maybe they're not meant to be teased apart. Maybe they actually work as a matrix, and attempts to deconstruct them, while useful, may actually forget or may not allow us to really see the big picture. It's sort of, the quantum physics versus Newtonian physics. I mean, can we actually distinguish the wave from the particle? And there's a practical aspect of it, because I want to just toss out that nobody's really talked about how do you influence this matrix. The only way nature knows how to influence this matrix is to stimulate the organ, not the nerves. The nerves are responding to what's happening in the organ. So maybe this quest for a nodal point, with all apologies to GSK, is not necessarily going to be successful. So we did an experiment where we took a liver, which is not generally regarded as being highly innervated, and we simply, and this is published by the way, we simply put electrodes in the parenchyma of the liver. We don't even know whether it was next to any nerves or not. And we stimulated it with some highly specific parameters, and we were able to attenuate diabetes in rats, both in type 1 and type 2. The point I'm making is we should not forget that modulation, electrical neuromodulation, is not simply at the nerves, and we should think about this matrix as a whole and how to influence that.
But isn't your matrix made up of excitable tissue?
Yeah, but there are some things about this, perhaps, that are not meant to work in isolation, can never work in isolation. Is the Heisenberg uncertainty principle. The moment you actually start observing it, you've distorted it. And so maybe we should, I mean, this, like I said, is being provocative. I just want to add that.
Yeah. Jay brought up again the issue of the ganglia. We've really not paid much attention to that except for the cardiac ganglia. And the ganglia, traditionally, we often thought as just relay stations and distribution centers. The central nervous system sends out relatively few fibers, even though vagus has 100,000 fibers. Most of them are afferent. But in the pelvic viscera, there may be 1,000 or so preganglionic fibers that come out and are distributed to 20,000 postganglionic neurons. But those ganglia have the ability to integrate input from sympathetic and parasympathetic pathways. They have the ability to modulate the activity coming from the spinal cord to the bladder or to the colon. So these ganglia are very important structures, and we shouldn't really ignore them in our goal to understand human physiology. There's not really very much known about the function of those ganglia. Excuse me. So I think I'm running out of time, and we need time to talk about technology. We had hear from Yvette, need for dissecting local reflex circuits. Yes, definitely. What happens in the organs? Even in the bladder, where we think it's relatively simple in comparison to the enteric nervous system, there may be local circuits mediated by intramural neurons and afferent pathways. Define the time course of vagal stimulation response in different parameters of stimulation. Okay. And Kevin, you're up.
All right. Grace and Jill will kill me if I don't go quickly. So I'm just going to go quickly through the summary of the technology points and then open it up for discussion. So if there's particularly, I guess, identify your particular issues related to technology, and if you don't see them up here, then you should bring them up, I think is how we should work this. Oh, I forgot. Yeah, we have these early vagal nerve stimulators. I'll just mention these, highlight them, not go through the details. Obviously, we talked a lot about the translation of animal models to human applications and whether the technologies that we're using will scale to humans or scale down to small animals, and there are some significant problems there. Selectivity is obviously the key for many of these technologies, and whether we can, this is an area, I guess, of big development, trying to identify ways and means of selectively activating the nerves. Sensing and sensors, again, that's a huge area, and it's got to be part of this program. Understanding, I think Chet just mentioned this one, understanding the effect and importance of all the different parameters, whether it's electrical parameters. It doesn't have to be electrical. It could be some of the other modalities as well. Device-drug combinations was already mentioned. Understanding how to customize and tune these systems for each individual and probably may require some adaptive systems, and we haven't talked about that a lot, but that's a part of a lot of existing technologies that are out there. Characterizing the technology to the same extent that you characterize the physiology. Some of these things, particularly the newer technologies, are not as off the shelf as we would like them to be. Access to the technology may be an issue, and that may be part of what needs to be in the SPARC program, ways to get access to these tools. There's, of course, not just the tools that might be used for clinical applications, but also the enabling tools, and I think we heard about that in the session just before this. So there's a number of pretty exciting tools that probably aren't going to be used in human applications, but certainly allow us to understand the physiology, and those are obviously critical. And I think the opportunity for cross-working fertilization between physiologists and engineers, maybe even training sessions and workshops, I think, at least I know I learned a lot sitting here. I have a whole bunch of grants now to go back and write. And people to work with. So I think these are really good opportunities, and probably worth continuing in some form or other. And I think I forgot. And then the data coordination panel, just some of the things from here. So exactly how we do the data coordination, combining data from multiple places. How do we get access to that data? How do we protect the IP and the early stage experiments? And is that what I just did? I guess there must be 2 copies of that slide. And then these were some of the questions on the data. Should we have a central place for the data? And how do we plan to combine mapping data, sketches, things like that, in one place? And I think that is it. I had a couple questions for discussion as well, but again, just really the point of this is to bring up from the field the ideas that aren't in this summary. But a few people have mentioned this. What would it take for a device approach to be something better than the option of last resort? Right now, most devices are that patients get them just before a surgeon is about to take out his scalpel and cut the nerve. They get a device. Why can't we do that earlier? What would it take? Partly it's because it's so invasive. Should we consider implanting, enhancing implanted systems? I mentioned this earlier with biomarker sensors that would give us some data. And this really goes back to understanding the relationship between the animal and human models. If we can start to collect some data from humans as a natural part of our treatment, because we added some sensors in there, to me, that seems like a great thing to do. And I think that is all there is. Yes. So the floor is open.
Can I just respond to I think it's Jay, you mentioned this question of an integrated system, and we're poking little parts of it. And how do you understand the big picture and also the individual trees? And I suppose it's really, perhaps it's obvious that we have a choice of resolutions, rather as in the times when the microscope and the telescope were both invented, and they expanded our understanding and our range of ability to observe. So, I mean, within the SPARC program, I imagine there's a range of resolutions at which we can work, and they're not mutually exclusive. The fact that you're studying a vesicle doesn't exclude the fact that you're studying a system, but we have to be clear about what resolution we're working at, and different techniques perhaps for the integrative kind of studies versus the very detailed studies.
So, Kevin, one technique that I haven't heard discussed at all today that I think would be valuable, so we stimulate the vagus nerve and we get 8 different diseases cured. And I think if we, the CFOS technique, It doesn't label every neuron that's been activated, but if you were to take the vagus stimulation at different points using different pulse duration, cycle duration, and just generate a map and says, okay, when we stimulate the vagus and we're looking at this end response, we get nucleus tractus solitarius, we get the parabrachial nucleus, those sorts of things. And again, that leads into my next comment is in the data accumulation, talking to most of the physiologists, a lot of the physiology data, it's just so capricious that you're not going to get common CDEs, elements. But I think in the anatomy, so looking at the nodose ganglion with the CART expression, those sorts of things, I think there a giant database would be very good because, we each look at 3 different transmitter co-localizations or receptor co-localizations, which organs they innervate. So I think there you could set up a spreadsheet where, the lab in St. Louis injected here and studied these 3 co-localizations, and eventually that kind of data would be searchable and valuable to say, okay, are there any neurons that are very specific to a population that co-express 5 different transmitters and have these 8 receptors? And then along with that, these CFOS maps. I think there again, there's not, you map where they are, and that would be something, okay, I stimulated this nerve at this rate and got this pattern. So those are my 2 comments. Thank you.
Jill and Grace, are you guys making notes for this? All right, good. So I don't have to remember these. Go ahead.
Quick question. I wanted this panel to discuss we were victims of uncustomizable technology. I just wanted to discuss that. Some clinical people talked about, not being able to change parameters once they implant the devices. I wanted to know from the community what they thought of highly customizable devices, implantable devices.
Kip, go ahead.
Really to emphasize that as well, I think a lot of times we're looking for long-term chronic data where through regulatory approval we have to lock in our systems. I think for all of this stuff, there's a lot more that can be done under NSR than we're actually doing right now. We're trying to take a locked-in system and get chronic data. There's a lot of opportunities here for all this community to have access to the vagal nerve if you're stimulating it for epilepsy, to look at it for other things as well, and to do short-term studies, even under 30-day, to get a lot of this data that you're talking about that we need, and to develop biomarkers in a way. I think we're thinking too far about the manufacturing and how to make these work for a year, when a lot of this stuff we can do and get a lot of information in 2 weeks.
Well, thinking about customizable devices brings back the question about parameters that we need for stimulation. And the provocative thought that I had is that maybe we don't need different parameters for stimulation. What we hear is different parameters work, and either we are very lucky to choose every time efficient parameters, or parameters do not matter as much. And we also hear that if we cut the nerve or block the conduction, we can get the same response as from stimulation. And the only explanation I see to this phenomenon that we are not really dealing with neuromodulation. We are doing with scrambling of the signaling that comes from the organ to CNS. So CNS does not really control anymore this organ is the way it was trying to control before. And if we are scrambling the signal, it does not matter much whether we see scramble is with 1 hertz, 2 hertz, 15 hertz stimulation, with short bursts or long bursts, we are scrambling. And again, we can block conduction instead of scrambling. The result will be the same because CNS will no longer receive the information about what the organ is doing. And there has been one remarkable exception to this seemingly universal rule, and the exception was today when the distal part of the cut nerve was stimulated to activate T cells in the spleen, but that's why it is an exception, because in this case, the nerve was used as, well, as a cow. We just milk it to produce neuromediator to activate T cells. There was no targeted connection of the nerve to cells in the target organ. So maybe we need to have a second look at what we call neuromodulation. It's not always a modulation. We're just interfering with normal function, and this normal function may actually be diseased. So by interfering with it, we can cure the disease or improve conditions. So it's not necessarily bad. Thank you.
Go ahead.
So I'd like to respond to that, and then I have another comment about your question.
Go ahead.
So I agree that if you take a mixed nerve, not only mixed because of the sizes or conduction velocities, or whether it's, but also because of its afferent or efferent, whether it's sympathetic or spinal or vagal, all together in that bundle, and you try to stimulate it, you're going to get some kind of garbled result. So maybe your scrambling theory is correct in that regard, but there are also specific examples where you can start at the periphery with well-defined parameters with opposite physiological effects and some set of parameters not having the desired therapeutic effect. So both may be true, but depends on where the site is and how specific the actual nerve that you're targeting may be. So if you're doing it with a mixed nerve, I think the chances of getting a scrambled message are more likely. I want to answer your question about what it would take for a device approach to become something better than the option of last resort. And this is not just simply technology or invasiveness. I think we have to factor in, and this is increasingly being done, perhaps was part of the decision behind why the FDA approved the last obesity device, is the patient's perspective. We are going to a patient-centric world, rightfully so, and we need to start taking into account patient choices. If you give, and that applies not just from a regulatory perspective, but also a reimbursement perspective. Nobody has taken into account on either side, coverage or regulatory, what the patient really wants to do. The patient has followed an algorithm that has been artificially created by us, which said, well, if you don't respond to surgery, then perhaps because of either regulatory issues or because of reimbursement issues. But if you give patients a choice and you say implanting a stimulator around a nerve early in the course of rheumatoid arthritis, to give that example, rather than subjecting you to multiple rounds of anti-TNF therapy, they may go for that, but their voice doesn't count. So as we develop new technologies and around new phenomenon, I think we must take into account the patient's perspectives early on, rather than that. And that will then, answer your question.
Right. Yeah, that's an excellent point. Brendan?
I wanted to reinforce your point about the importance of engineers and biologists to work together on these difficult issues. And that's especially true in organ systems and diseases where devices have not been widely embraced, and the airways would be one obvious example of that. Because I think one of the things that we want to avoid is to lose the connection between efficacy and mechanism. Because I don't think we want to be in the position where we say, and then something magic happens and the patient felt better. I think that would be a bad outcome, and it would limit how widely these technologies would be embraced in new diseases.
Yeah, I think we have to close. Can we take one last question?
Take one if it's
All right, Joe will allow one last question.
I'll make it, well, it's actually a comment, and for those of you who haven't followed the renal denervation world and treating hypertension, it relates to what we're talking about here. So that was based on the idea that nerves were overactive to the kidney, and that was the cause. You could get to the renal arteries and ablate the nerves. First two clinical trials were hugely successful. A one-time treatment, 3 years later, people's blood pressure was still down. Then when they did the clinical trial here in the US, it didn't reach its efficacy endpoint. Two major problems were there's no diagnosis for who should get the treatment, so maybe it's not the same patients. The other is you don't know if you did it properly. So we're still trying to figure out what happened there. So that comes back to the question I had for you this morning. I think you have to be very careful about knowing that the device is actually working, especially if the outcome is slow to see. Maybe that's not the best last thing to say, but.
I would like to take a 1 minute or 2 minutes, actually as long as he wants, for Dr. Anderson to come up and and speak to you for a moment. And then I think all of us would like to take a moment to express our appreciation to this group and to the chairs. So.
Okay, well, thanks, Jill. I'll be very brief, and I'll just stay in my seat. How's
okay.
that? I did want to add my thanks for everyone's participation, for bringing your expertise and your ideas. It's really been a successful, thought-provoking meeting. I just want to reflect that we started this with really a single, simple, and compelling goal, and that was to identify the research that's needed to catalyze the field of neuromodulation. And it, as often happens when you get a lot of smart, informed people together, it blows up, and there's just lots of different potential directions. But as a Common Fund program, we're going to have to refocus on what are the highest priority and highest impact deliverables we can produce that'll move this field forward, and with a finite period of time. That doesn't mean it's the end of funding for the field. It means that the institutes and other parts of the world will pick up the field, but we need to identify what's going to move it forward quickly. So NIH is, this will be a challenge because we need to keep technology folks together with biology, and there'll be it'll be very fluid in the opportunities we can take. So NIH is going to take a very unusual management approach to this by using a project manager. Similar to DARPA's approach, this person will have a high degree of authority both in identifying directions and in the review process and in management. And we need a very high quality and informed person. So if any of you know someone with these types of leadership skills and the scientific content expert and the Rolodex that's going to be needed to do this, please contact me. I'll serve as the point of contact for that, and my address is in our book here. So thank you again.
Okay, thanks, Jim. And I would like to add a couple of words of thanks of my own. So first, thank you to the speakers and to the chairs and discussion leaders who have served us through this meeting. I also want to mention that although I had to be timekeeper and Chet very gracefully accepted my timekeeping hurry-up, he and Terry Powley answered my phone calls when I called and said, this is something that's going to happen on a short timeline, and I need help and expert advice on putting this workshop together. And I think that Grace also contacted some people in that regard. And people have made themselves available for phone calls, and Chet and Terry did that multiple times over the course of weeks. And I want to especially thank them. I also want to say thanks to Christina Falk, who did the yeoman's work on getting this thing going from the administrative point of view and designing programs and things like that. So thank you to her and to the whole Spark team for their input, and to the community and all the people here who have given us a great start on thinking about how to figure out what the priorities for Spark will be. So I don't know, Grace, if you want to say anything further.
I just want to echo Jill's thanks, and especially to the technology speakers. So basically, after Jill rounded up the biology speakers, I was given the task to match them with technology speakers on short time periods. So thank you all for the technology speakers for responding so quickly and coming to the workshop. Very glad that you were here to provide the responses that we need. I also wanted to let you know that the video cast will be archived along with the slides that are provided by the speakers. So you can check our website for that information. And as well, don't forget about the March 5th informational telephone conference for our First Spark initiative, the U18 FOA. So there we can answer any further questions you might have on this very first initiative to promote technology. I think many people were confused that it was to understand biology, but it's actually to promote new technologies. So with that, I guess we'll adjourn the meeting, and thank you all for coming, and safe travels back in the snowy weather. Thanks.